Yes, Shivangi.
Yes, sir.
Now we can start, no? Hello, Gurjit. You are muted.
Can you see me?
Hello.
Yes.
Yeah. Do you see my presentation?
Yes.
Oh, okay.
So can I start?
Yes.
A very warm good morning to one and all present over here. Our speakers, our honorable speakers, teachers, students, and all the faculty members. I, on behalf of Institute Innovation Council, IIC Galgotias University, invites all of you to international seminar on innovative strategy for neuroprotection to celebrate National Education Day. The speaker for today are Dr. Sumedha Mukherjee, Mr. Mohammed Salman, Dr. Gurjit Singh, and Dr. Dhanananajay Kumar. Before letting our speakers tell us more about neuroprotection, let me introduce you about what IIC Galgotias University basically is. IIC Galgotias University established as per norms of Ministry of Education Innovation Cell in academic year 2018 to 2019. In 2020-21, Galgotias University recognized in the band Excellent under category University and deemed to be university in utter ranking of institutions on innovation achievement area. In 2022, Galgotias University received Certificate of Appreciation by Intellectual Property Office, Delhi, Government of India. I'm very proud to say that we have 13 IIC officers, 20 innovation ambassadors, 19 faculty innovation coordinators, 19 faculty entrepreneurship coordinators. The Galgotias University has set up its incubation center as Galgotias Incubation Center for Research Innovation Startup, GIC Rise in 2020. Currently, GIC Rise has 30 startups in the pre-incubation stage. Among them, 19 are registered on MSME, 8 startups are in the incubation stage and are registered as private limited LLP entity. Number of graduated startups are 8. We have 100 plus innovation cell student members. We have organized 31 events in the academic year 2020 and 2021. We have organized 55 events in the academic year 2021 to 2022. We have published, filed, and granted of around 960 plus patents, and we have 100 plus alumni entrepreneurs. Moving forward, now I would like to invite our speaker, Dr. Gurjit Singh, Research Associate, University of Illinois, Chicago, USA. Currently, Dr. Gurjit Singh is working as Research Associate in the University of Illinois, Chicago, USA. He has received PhD degree from Guru Nanak Dev University, Amritsar. Previously, he has worked as marketing executive in Elmic Pharmaceutical Private Limited. He has qualified graduate pharmacy aptitude test in 2013. He has received various awards, including Rajiv Gandhi National Fellowship Award and Travel Award from International Society for Neurochemistry. He has published more than 15 research papers in reputed SCI index journals and presented several papers and posters in national and international conferences. His research areas are pain, inflammation, depression. He's active member of Society of Neuroscience, Canadian Association for Neuroscience, and International Society of Neurochemistry. Sir, I will present the stage to you and thank you.
Mm.
Thank you. Thank you so much. Thank you, Gurjit, for accepting our kind request to deliver your invited talk. Over to you.
Thank you so much, Gaurav. Yeah. Thank you so much, Shivani. Yeah. May I start here?
Yes, yes.
Yeah. Good morning, everyone. Yeah. Today's my topic of the presentation is the osteoarthritis and the therapeutic targets. So what is the osteoarthritis and what is osteoarthritic pains? Osteoarthritis is a cartilage degeneration disorders. It's main like degeneration of articular cartilage. Where is occur osteoarthritis? Various our joints, in body, our joints, it causes osteoarthritis. First, knee joints, hip, ankle, toe, lumbar disorder, neck, shoulder, finger, and temporomandibular joints. So why it's OA is a more painful condition affecting pupils. About 32 million Americans affecting for osteoarthritis, and its economy burden is more than 2000 billions. So that's the reason 50% of OA patients are treated with opioids, and many have become addicted contributor opioid crises. So it is an urgent unmet need to improve treat strategy to manage chronic joint pain and associated depression simultaneously. So that's the reason why we get osteoarthritis. OA risk factors. Whereas there is a various risk factor of osteoarthritis. First is injury due to a military and some sports persons. And second thing, aging. Aging is a main cause of osteoarthritis. When is age, the cartilage degeneration is more due to aging and genetic factors, smoking, obesity, and alcohol consumptions. So that's where is there is a lot of treatment options for osteoarthritis. There is two types of options like non-surgical options and surgical options. So non-surgical options like we use already painkillers like aspirin, buprenorphine, and other NSAIDs. Some nutrition supplements, glucosamine, chondroitite sulfates. And now the strategy to use for osteoarthritis is mainly intra-articular injections like steroid, hyaluronic acid, lubricant, and platelet-rich plasma injections. Some other therapies like exercise, yoga, weight loss, and acupuncture. Now the current strategy, the researchers are working on a stem cell therapy. This therapy used directly stem cell inject on the knee joints. And that other surgical options like for minima invasive to joint replacement surgery, no further option knee, hip, and replacement surgeries. Surgery won't be a granted pain-free, and there is a no cure of OA. So that's the reason is cause of is need to improve and prevent the osteoarthritis. We need better strategies. Why is osteoarthritis causing? The osteoarthritis is age-related cartilage degenerations. OA is a whole joint. This is a whole joint of knee joints. We take small portion of these knee joints. Like this one is a cartilage section and this one is a synovium. Some main things, degeneration of cartilage is causing of osteoarthritis. In degeneration of cartilage, it release lot of cytokines and nerve growth factor in synovial fluids. Synovial fluids. That pain transmission not involved from cartilage because there is no nerve formation in cartilage. The pain transmission only on synovial fluids. So that's the reason the pain is only transmitted by the surrounding tissue of the joints, not from cartilage. So centralization of nociceptive pain in the knee joints, how it's cause centralization of pains when the knee joint, when injury in knee joint is peripheral injuries, the primary nociceptor activates C fibers and A delta fibers activate, then it causes signal to the DRG, and DRG regulate the lot of molecules like CGRP, substance P, NGF, VGF, VGF receptor 1, neuropeptides also release in a DRG, and they signal to CNS and then enhance excitability of neurons and signal to brain and causes of pains. Why pain is a transmission process from peripheral tissues, peripheral sensory neurons, and then peripheral sensory neuron to central nervous system. The amplification of pain is less evocative, is called as central pain sensitizations. So that's the pain. There is a 4 type of pains, like nociceptive pains is reversible pains when it's injury and you cured it, that is called a reversible nociceptive pains. Second is a neuropathic pain. It's irreversible, caused by nerve damage, nerve compressions. So it's irreversible. It's not cured by any medicines. So third one is a chronic inflammatory pains. It's a reversible cause caused by chronic inflammation. Fourth one is a mixed pain. Mixed pain is all 3 type of pains, like a nociceptive, neuropathic, inflammatory factors. OA is a mixed type of pains. It's induced by nociception pain, overlap with etiology of neuropathy, inflammatory pain mechanism. So that's the reason OA is a mixed type of pains. So next target is why targeting pain specific to OA is important. So that's the reason nociceptive, inflammatory, and neuropathic pains may result from diverse mechanisms. Hence, the intrinsic unrealistic to develop a drug that attack the root cause of all these pains. For one example, pathology of diabetic neuropathy is due to damage of nerve that disappear, compared to the OA pain due to much nerve in growth formation, because that reason in OA cases the nerve in growth formation in a synovium, and it causes signal to nerve to the DRG from peripheral nervous system and peripheral nervous system to centralizations. The rationale of treatment of pain in OA required identifying the primary OA specific pain target to control OA associated pains. So in this study, our main goal is a mechanism based identification of OA specific targets and development of OA disease modifying drug specificities. We are working on 2 targets, NGF and its a receptor TrkA, and second is a VGF and its receptor is VGF receptors. VGF receptor is 2 type of receptors, VGF receptor 1 and VGF receptor 2. VGF receptor 1 is involved in both in cartilage degeneration and pain perceptions, but VGF receptor 1 is involved only in cartilage degenerations. So for we concluded that NGF is role in osteoarthritis or not. So then we just study on retrograde transportation of NGF is accelerated on OA. We hypothesis that in symptomatic OA overexpressed synovium, NGF could be retrograde transportation to the DRG through trachea exciting a afferent sensory nerve terminus. This may stimulate sensory neurons to promote axonal outgrowth and pain. So in this case, the peripheral injury in the joints, then fibroblasts like synovial cells is produce lot of spores, inflammatory cytokines, bradykines, and chemokines, and they release NGF, and they cause nerve outgrowth formation in a synovium. Then NGF bind with the trachea, and they went to the retrograde transposition in the DRG, so that the transcription of molecules of neuronal outgrowth pain to peripheral to DRG sensory neurons. So increase the retrograde transportation of NGF is in OA. Why? The introduction of induction of knee OA when we inject inject biotin ligand NGF in advanced OA, we harvest DRG after 3 days. We are using mainly lumber 3 to lumber 5. And then we check biotin immunoreactivity with streptavidin alexaflor for 18 green signalings. So in this case, we found that in advanced OA, in lumber 3, lumber 4, the green signal shows that the binding of biotin and NGF is more expression in advanced OA. So that's why we are using ligand biotin with NGF, because there are in OA conditions, the lot of NGF production in a sensory neuron DRG is NGF, so that we differentiate from retrograde study from these NGF, we ligand with biotin, that ligand with biotin, that is biotin ligand conjugated with NGF, so that the detection of biotin immunoreactivity in a lumbar in DRG from OA joints. So that's in retrograde transportation of biotin conjugated NGF from knee joint to DRG in OA. It suggests that abundantly sensory nerves in OA knee joint facilitate nerve growth retrograde transportations. So in this case, NGF and trachea is vice versa to VGF. If NGF is increased, VGF also increase. Then the stimulation of lot of inflammatory cytokines, nerve growth factors, substance P's in OA disease. So next we done this study, NGF role of loss of PKC delta prevent cartilage degeneration in joints, but accelerate hyperalgesia in experimental model in osteoarthritis. What is the PKC delta role of PKC delta in osteoarthritis? PKC delta is a key mediator of cartilage degenerations. So in this study, we are using a transgenic mice. We are using a PKC delta rosacea. It means a knockout of PKC delta in whole mice, and second mouse we have using a PKC delta in sensory neuron. It means a PKC delta knockout only in sensitive neurons like DRG. So in PKC delta knockout whole body mouse is the PKC delta knockout in cartilage and sensory neuron both. So we comparison in PMM surgery is a wild type mice, OA mice, compared with the PKC delta knockout whole mouse. We shown that the cartilage protection in PKC delta whole mice, but in sensory neurons, specific deletion of PKC delta in sensory neuron, it did not affect on protection on cartilage degeneration. So then we compare why it's protect cartilage PKC delta mice in OA conditions. So that we did is MMP13 enzymes. MMP13 is a catabolic enzyme, collagenase 3. It is a main enzyme for cartilage degeneration. When we immunostaining of these enzymes from PMM wild type OA mouse with rosacea whole knockout mice, we shown that the increased expression of MMP13 in OA mouse models. But when we treated when we are using PKC delta knockout whole body mouse, it reduced the expression of MMP13. So that's the reason it protects the cartilage degeneration. In other cases, see, PKC delta knockout only in sensory neurons, not in a cartilage. It did not protect the cartilage degeneration and increase the expression level of MMP13. So we found that the cartilage protection of PKC delta knockout mouse is due to a reduced expression of MMP13 in OA mouse. So next, we found increase PKC delta mouse protect the cartilage but increase the pain perceptions. So we found that why it's increase the perception, reduce the cartilage protection. So we found that in synovium, increase the expression level of NGF in PKC delta knockout mouse compared to wild type OA mouse, and also shown in in PKC delta knockout in sensory neuron is increase the expression level as compared to wild type mice. So that we found that the expression of peak NGF and TRK is more in PKC delta knockout mouse in as compared to OA wild type mice. So in this case, PKC delta regulate the NGF. If PKC delta present, the NGF level is reduced. When PKC delta knockout in synovium and whole body, the expression level of NGF also reduce. So we found that the NGF level is main cause of pain when we deletion of and PKC delta in mouse. So next we found that in DRG, we are using a co-localization of NGF in DRG. We are using a neuronal antibody with NGF. In this case, co-localization is yellow spots shown that the NGF expression in neuronal ends. So we compare our NGF level with OA wild type mice with PKC delta rosacea mice. The expression level of NGF in neuron is increased in both PKC delta rosacea and PKC delta sensory neuron. Deletion mice is increased the expression of NGF as compared to OA wild type mice. So that in another case, TRK is also increased the expression of TRK in the OA wild type and OA PKC delta mice and PKC knockout in sensory neuron mice. So this pathway is shown that the NGF is the main role to regulate the pain in PKC delta knockout mice in sensory neurons, because when we knock out PKC delta knockout in sensory neurons, it induces the level of NGF. When it's induced the level of NGF, it causes induce the pain perception in peripheral system and then cause to centralization of the pains. So in these studies, we have concluded that our results shown that the genetic loss of PKC delta proteins in the joints to protect cartilage from degeneration by inhibiting the catabolic enzyme like MMP13, and loss of PKC delta in sensory neuron exacerbate hyperalgesia, especially by overacting of NGF-TRK signaling pathway in our preclinical osteoarthritic models. There is some limitation of these studies because PKC delta in knockout mice protect the cartilage in osteoarthritis, but it induces the pain. So that's the reason targeting PKC delta will not be ideal for relieving OA pain. Instead, inhibiting NGF-TRK may represent an effective treatment strategy to the mitigation of the OA association pain. So that's the reason we did not continue this study. So thank you so much for.
Okay. Dear participant, if you have any query, please ask. The now session is open for the question answer session. So related to the osteoarthritis and their targets, pain, you can ask your question to the speaker. Any question? Are you able to unmute yourself?
I have a question.
Yes.
What is the main cause of OA?
The main cause of osteoarthritis is cartilage degenerations. Like when your cartilage degenerations, then the more nerve growth formation in a joint, and it causes neuronal outgrowth formation in a joints, and it causes a pain and cartilage degenerations.
What causes the cartilage to degenerate? Is it the food that we eat or?
Yeah, it's depends on various mechanisms, what we eat, what we drink, like a food food like obesity is a main cause of osteoarthritis. When your weight is increased, the more weight on your knees, then it's your both bone like tibia and fibulas compressed, and your cartilage degeneration more severity. And when you use alcohols like smokings, they induce the expression level of PKC delta NF kappa B in a cartilage, and it induce the degeneration of cartilage. So that will cause the osteoarthritis.
Thank you.
Also, there is a reduction in the amount of the synovial fluid there, but in this case, the pro-inflammatory cytokines are triggered due to the what to say, the friction between the bones, right? Due to reduction in the synovial fluid, right? In this case, so
No. Yeah, it's
osteoarthritis.
cartilage degeneration is another factor. The pain perception is another factor. No.
Yeah. So what I want to ask here, that what is the role of the pro-inflammatory cytokines build up here? How do they trigger the how do they trigger this whole process?
Yeah, role of pro-inflammatory cytokines when the cartilage degeneration, it induce the expression level of pro-inflammatory cytokines cartilage to synovium. When it's cartilage to synovium, then it's triggered to neovascularization and release NGF and VGF in growth formation in synovium, and it causes a sensation of pains. The cartilage degeneration is only affected by MMP13 levels because it's a catabolic, yeah, collagenase 3 enzymes. So when collagenase 3 enzyme is increased, the cartilage degenerations, and it produce lot of cytokines, VGF, NGF production in synovium.
Okay. Yeah, I was missing this link between the two. I know that pro-inflammatory cytokines are stimulated, they are overexpressed, but I was missing the link between the cartilage degeneration and this. Thank you.
Okay, any other question?
What are the early signs of this during like it's happening or not? What are the signs and how we can recognize it in early stages?
We did not recognize this early and advanced way because some patients like symptomatic pains, some like asymptomatic pains. So we just found when we know when we have pain in a joint, then we will go to a doctor and just X-rays when we know. But otherwise, in preliminary, we did not found any osteoarthritis. But in some patients, when it's severe osteoarthritis, but it did not show any pains, it's a normal fact. It's only cause when you regular checkup to a doctor and something.
Any other question, or we should proceed further?
No question, sir.
So now I would like to request our speaker, Dr. Gurjit, to kindly accept our e-memento as a token of love on behalf of IIC Galgotias University.
Thank you so much, Gaurav.
Thank you very much, sir, for the nice presentation, for the nice information related to osteoarthritis as well as pain receptors. Thank you. Thank you so much, sir.
Thank you so much.
Our next speaker has joined. Dr. Salman.
Our next speaker for today is Dr. Mohammed Salman. He is currently working as postdoctoral researcher in the University of Tennessee Health Science Center, USA. He has received PhD degree from Hamdard University, New Delhi, India. His research areas are in traumatic brain injury and stroke surgery, having expertise in measures of immunocytochemical protein analysis, mitochondrial biology, oxidative stress, immunohistochemistry, and behavioral techniques. He has years of experience in various models of neurodegenerative disease models such as Alzheimer's disease, Huntington's disease, Parkinson's disease, neurotrauma, and neurobehavioral studies. Published more than 16 research articles in the field of neuroscience and neurodegeneration in reputed SCI index journals. He has received various fellowships, including JRF and SRF from University Grant Commission, as well as JRF and SRF from ICMR, Ministry of Health and Family Welfare, Government of India. He has also received international travel grants from Ministry of Science and Technology, Government of India, international travel grant from Indian Council of Medical Research, Government of India, and Best Poster Award in the International Brain Research Organization, Banasthali University. He is member of editorial board of Journal of Toxicology, American Journal of Psychiatry and Neuroscience, Frontiers in Neuroscience, and Saudi Pharmaceutical Journal. Over to you, sir. Thank you for attending.
Thank you so much, Dr. Gaurav, to invitation for me. So I'm going to present. Should I start?
Yes, sir. Yes, sir. You can start.
Okay.
Thank you. Thank you so much.
Yeah, thank you so much. Okay, so you're welcome. And my topic is today pathophysiology of ischemic stroke, present status and future perspectives. So nowadays, I mean that this is the second cause for the morbidity and mortality, and that in the old and young as well as population. So why it's not changing? Yeah.
Yes,
So
slide is
in
changing.
this, yeah, in this presentation, we'll cover two objectives. One is the complete focus on signs and symptoms of an ischemic stroke, and second is the Murine model of ischemic stroke, and third we'll cover the photothrombotic model. Because today I'm going to present the photothrombotic model because all the models, including the embolic stroke, thrombosis, and other ischemic strokes, they have the higher mortality rate. So we are targeting how to reduce the mortality, and we can go for the short-term and the long-term effects of the ischemic stroke with any drug respects. So introduction. Nowadays, the human population is suffering from the disease and the health issues. So that can be communicable diseases, non-communicable diseases, different types of injuries. In communicable diseases, we will know about the COVID-19, Ebola, flu, hepatitis, tuberculosis, etc. For non-communicable diseases such as cancer, chronic lung diseases, diabetes, Alzheimer's, Parkinson's, etc. For different types of injuries, you know, the brain injury, spinal cord injury, different types of trauma, etc. So I'm going to highlight my research work on the stroke. So what is the ischemic? What is the stroke? So this is the basic question. Okay, what's the stroke and symptoms? So strokes occurs when there is interruption in blood supply, or we can say the blockage or bleeding inside the brain to an area in the brain leading to the brain damage, tissue damage. So how many types of the strokes in human condition? So there are three types of a stroke. One is ischemic, which caused by a blockage in the blood vessels to the brain. It categorized into two types. One is embolic, second is thrombotic. So embolic is travel. For example, if the blood clot is formed elsewhere and then travels to the brain and blocks the artery and causes the ischemic condition. Thrombotic is due to the plaque inside the artery and then blood clot is stuck over there and then reduce the blood flow inside the brain. So over here in this picture, you will see the blood clot is stop the blood flow to an area of the blood to the brain. Second is the hemorrhagic stroke. It's caused by the burst or leaking blood vessels in the brain. These are the two types of, one is the intracerebral hemorrhage and subarachnoid hemorrhage. So you will see in this picture, it's the leakage of the blood into the brain and develops the uh, less of blood flow inside the brain. So third is the transient ischemic attacks. It's called mini stroke, resolves within 24 hours, but it can happen again and again. So this is the most causes in the young generation for the ischemic stroke. So what's the epidemiology? I found a few literature about the US and the world and India. So 1 in 6 die of the stomach stroke in lifetime. So overall, 20 million people suffers from a stroke every year. It causes 5 million deaths. 5 million people are rendered disabled because of the stroke. And 1 in 6 persons get a stroke in his or her lifetime. So if we talk about the India, 1.5 million people suffers from a stroke every year, and 3 to 400 Indians suffers from the ischemic stroke every day. Strokes causes more death in the country than malaria, tuberculosis, and HIV combined. So stroke now affecting more younger people. Global burden could be double in 2030. So why the younger people is more prominent for the ischemic stroke? I will talk in next slide. So nearly half of all US adults have some form of cardiovascular diseases. Let's talk about the India. Did you know stroke facts? On an average in India, a stroke occurs every 40 seconds, and every 4 minutes, someone dies of the stroke. So half of all strokes in India can be attributed to high blood pressure. That's the main cause of the stroke. 1/4 of all stroke in India occurs in people who have already had a small stroke. That is called the small stroke. That's the, I mean, it can resolve within 24 hours, but it can happen again. So according to the news reporter, I don't know, but exactly, be aware youngsters too are at a higher risk of stroke due to the air pollution. It's this small study. So what's the sign and symptoms of the ischemic stroke? So first is the headache. Second is mental changes, confusion, dystonia, memory impairment, aphasia, vomiting, nausea, coughing, swelling, relaxed seizures, and emotional ability, visual changes, respiratory problem, practical deficit, and hypertension. So how we can identify that? I mean, someone is suffering from the stroke, or it's the initial phase of the stroke. So it's called the FAST. F stand for the face. If you will ask the pupil of some a person who is, I mean, feeling uncomfortable, so you will ask for the smile. So they cannot smile, they will drop one side of the face. And arm, A stand for arm. So if you will ask to lift up something at the both arms at a time, so they cannot hold one arm, so it will go one down and one is up. So there's the symptoms. And as for speech, if you will ask to person for the prephrase, read for the prephrase, for example, would you like to eat something, so they will not comfort to say exactly. And time, T for time. Time is most important, because time is uh, it's depend on the ischemic condition. So again, it's replaced by the faster. So first, previous is the fast, now it's replaced by the faster. F for face again, F for arms, S for stability. So if you will ask to the patient, they could not walk properly. And the talking again, and eyes, so they have the blur vision. And react. React means if you will find these symptoms, so you should call to the ambulance or emergency call, something like that. So in United States, it's 911. So the people generally call to the 911, then the ambulance will care to the patient. So what's the risk factor of the ischemic stroke? So there are the two types of the risk factors. One is non-modifiable, and other is the modifiable. So age, family history, previous stroke, or gender, they cannot be modified, but high cholesterol, diabetes, high blood pressure, hormonal consumption, obesity, inactivity, alcohol intake, and substance abuse, smoking, they can be reduced. So how to diagnose of a stroke? So there are different types of method to diagnose the stroke, but it depends on the patient condition. So CT scan, physical examination, several angiogram, MRI, echocardiogram. These are the few techniques that we can detect the stroke. I mean, someone is suffering from the stroke, but it's the secondary. First is the fast and faster. So what is the pathophysiology of ischemic stroke? Once a clot is stopped the blood supply to the a specific region in the brain, so it causes vascular occlusion. It's reduce the cerebral blood flow, and then if the blood flow is reduced in the brain, a specific part of a brain, so it also reduce the oxygen glucose level, which halt the cellular metabolism and again energy production process, and alters of the cell membranes finally leads to the necrosis, which is the cell death, and apoptosis is the programmed cell death, and finally the neural cell death. So basic is that ischemic stroke clots obstruct the blood flow and hemorrhagic stroke, bleeding around the brain region. So they activates different types of events inside the brain: excitotoxicity, neurochemical injury, neuroinflammation, blood-brain disruption, interruption of the blood flow. So these events further activates the calcium ion and sodium ion flux, oxidative stress, microglial activation, and the increase in incremental pressure, and also depolarize the membrane and free radicals, finally leads to the cell death and cerebral edema and cerebral damage. So what's the current scenario for the treatment of ischemic stroke? So basically, one is the most important drug, which is approved by the FDA, is recombinant TPA, tissue plasminogen activator. So we'll focus on the two types of drug. One is the pharmacological and second is the non-pharmacological. So therapeutic intervention is the pharmacological intervention, so which is thrombolysis, TPA, approved by the FDA, but TPA has the side effect because after it's a short time window. So once a person is got the stroke, for example, at the 1 p.m., so we should note the time as mentioned in the uh, fast and faster. So we cannot give after the 6 hours of the stroke TPA. So that's the disadvantage of the TPA. So second is the mechanical thrombectomy, anticoagulant agents, antiplatelet agents, and neurotherapeutic strategies. As the mentioned, I mean, there's the title of this invited talk is the neurotherapeutic strategies. So I will discuss what's the important of these and what is the key factors included in the neurotherapeutic strategies. So non-pharmacological interventions, for example, yoga. So it's the regular the people do in the later age and the young age: exercise, meditation, and physiotherapy. So this is the a picture on your left side is the stroke. The first, I mean, the stroke happened in the early time point. For example, it's the 30 minutes. So you will see the yellow arrow is indicating ischemic core at the black dot, and it's the penumbra is indicating by the red arrow. This circle is the penumbra. So we cannot protect the ischemic core, but we can protect by the neuroprotective or neurostrategic agents these penumbra core. So untreated patient loses approximately 2 million neurons every minute in the ischemic area. Once the patient treated with the TPA therapy or other prominent therapy, so the penumbra is reduced by time, and then reperfusion offers the potential to reduce the extent of ischemic injury. That's mean it's important the time point in the ischemic stroke condition. So if the patient get the treatment in early stage, so that we can protect the brain from the neural damage by penumbra or ischemic core. So what's the neurotherapeutic strategies? So designed to save penumbra. So you will see here it's the core in the red color. It's called the ischemic core, where the blood leakage or the blood vessels stopped, and there is no more enough the blood flow, inside the brain, and this is the green color is showing the penumbra. So most of the neurotherapeutic studies target the penumbra, how to protect the penumbra. So for example, neuropathic targets is that inflammatory marker. So we use the anti-inflammatory drugs to reduce the inflammation, and calcium ion channel blockers. They reduce the calcium ion inside the mitochondria, and then they can reduce the mitochondrial mediated cell death, glutamate receptor, free radicals, for example, antioxidant, nitric oxides, protease and apoptotic pathways molecules, for example, BCL2 promoters, caspase, etc. And nowadays, people are targeting NLRP3 targets too. So animal models of the ischemic stroke. So in our lab, we are working on the in vitro, in vivo, and in vitro as well as, but here I'm going to describe the in vivo model. So ischemic stroke is divided into two models. One is ischemic stroke and the hemorrhagic stroke. So ischemic stroke further divided into two categories. One is global cerebral ischemia, and second is the focal cerebral ischemia. Global cerebral ischemia further divided into decapitation, cardiac arrest, systemic hypertension, and hypoxia, four vessels occlusion, two vessels occlusion. However, the focal cerebral ischemia divided into requiring craniotomy and non-requiring craniotomy. So in the requiring craniotomy include the surgical. So in this, we open the skull. The diagram is not showing over here. So it's the we open the little skull, and then we find out the where is the middle cerebral artery. MCA stand for the middle cerebral artery, and we cauterize the artery and we stop the blood flow. So for, and the next is the non-requiring craniotomy, in which the embolic stroke, interluminal suture, MCO model, stroke photothrombosis model, and endolethal one induced model. In embolic stroke, we prepare the clot inside a tube, and then we insert the blood clot through the uh, tube, with the help of middle cerebral occlusion. In interluminal sutures, we use the sutures, the basic model, and then we just insert the suture inside the artery and we block the blood flow for few time window. For example, 1 hour, 30 minutes, or 2 hours. So, and photothrombosis model, we inject the Ross Bengal dye and then we target the artery inside the brain, and then we focus on the head, and then we illuminate the light to the brain, and then we occlude the artery at a particular space. And hemorrhagic stroke is autologous blood infusion model and collagenase induced model, ICH. So, okay. Photothrombosis model. Photothrombosis model is a non-conical ischemic model. It does not occlude or break only one artery. It occlude so many arteries because we are focusing on the light. I will discuss in the next slide. One artery, as it usually happens in human stroke. So it is little different from the human stroke. So induces lesions in more superficial vessels, which results in selective interruption of blood flow in the areas of exposed light. So we use the Ross Bengal dye because it's the illuminated at the appropriate wavelength of 575 by a gold light source. It releases energy to oxygen molecules, which in turn generate a large amount of highly reactive singlet oxygen product. These oxygen intermediates induce endothelial cells membrane peroxidation, leading to platelet adhesion and aggregation. So for the confirmation of this photothrombotic model, so we confirmed by the TDC staining. So we performed one experiment in which we used the male C57BL6 male mice, 14 weeks old, and then we divided into two groups. One is the sham. Sham, we just expose the skull, not the light and the Ross Bengal. And second is the vehicle in which we induce the photothrombotic stroke. So we subdivided into two studies. One is short-term study, second is the long-term study. For the short-term study, we trained the animal for the rotor rod before the 5 days For example, if we're going to the stroke today, so we train the animal before the 5 days, and then we induce the stroke at the 0 day, and then we perform the rotor rod at 72 hours. And then we collect the brain for infarction volume, brain edema, hemoglobin, and confocal microscopy for the GFP and IBA1 proteins. So in the second study, it's a long-term study. So we induce the photothrombotic stroke, and then we perform the novel objective recognition test at 16 day after the stroke and 21 days to 30 for the Morris Water maze. So novel objective recognition test we performed for the short-term memory, and Morris Water maze for the long-term memory cognition or cognitive impairment. So this is the mouse brain image. So here we induce the stroke. So this is the MCA artery. It's coming down towards the brain, and we expose with the green laser light for the 5-minute exposure. So after the 5-minute exposure, we stitch the animal and keep back to the home cage. So this is the light source where we exposed. This is the exposed area of the brain, and after the exposing, so we confirm with the TTC standing after 72 hours for the thrombotic lesion. So this is not actual image, it's the internet image. I forgot to put the reference. So we perform the rotor rod for the motor coordination, because the stroke patients have the difficulties to the motor coordinate. They cannot walk properly. So we use this experiment for the motor coordination. So we put the animal on the rotating rod. So the maximum speed is 15 RPM for the 5 minutes. So we measure the time how one animal is taking, standing on the rotating rod or spending the more time on the rotating rod. So we found that the stroke animal fall down before the time point we set at the 300 seconds. So we check again the body weight, because the body weight is the primary sign of the brain injury. So we found that these animals of the photothrombotic stroke, they showed the reduction in the body weight. So this is the TTC standing. I'm talking about this one. We confirm this model by the TTC. So this is the sham. There's the nothing and no stroke. So these first we perfuse the animal, and then we collect the brain and we cut into the slices, different slices with the help of brain matrix and the both groups, sham and the vehicle, and then we stand with the TTC 1% TTC solution for 10 minutes at 37 centigrade. And then this is the dye. It's the tetrazolium dye. So it's react, I mean the SDES, succinate dehydrogenase, which presents in the mitochondria. It's reduced the dye into the red color. So where the SDS is present, it's showing the red color. Where it is not present, is showing the white color. So white color indicates with the damage, brain damage. So it's called an infarction volume. So we calculated with the help of image, and then we plot a graph. So we found that there is no infarction in the sham, but it's a high infarction in the vehicle group. Again, we check the ipsilateral edema. So after the stroke or the brain cell death, so the ICP is increased, intracranial pressure is increased. So we calculated the ipsilateral edema. That's mean there are two parts of the brain. One is the left hemisphere and the second is the right hemisphere. So if you will see, there's the white part here and there's the whole red part on the other side. So this is called the contra and this is called the ipsilateral, because there is no stroke or no signs of this cell death or the brain damage in the contralateral. So we include the ipsilateral, and we measure with the help of with the image software. We calculated with the image software, and then we found is the higher brain edema. Again, we check the hemoglobin content because after the stroke in thrombotic and other types of stroke, we saw a red color after the perfusion of mouse brain. Over here we calculated the hemoglobin, because if some blood is leaking inside the brain, so it will not wash out with the perfusion. So we check the content of hemoglobin concentration. So it's the higher in vehicle. We checked few confocal, few proteins with the confocal microscopy, for example, astrocytes activation. So after the stroke, astrocytes activated. So these are the sham, and these slides, brain slides from the vehicle. So here is the very less green color. So it is the GFAP protein, which is a marker of astrocytes activation, which help the higher expression of or higher activated astrocytes uh, increase the inflammation. So these are the blue color DAPI, which is the normally we stand with the nucleus. So over here you will see, we can see that the green color is high as compared with the sham. So that's mean ischemic stroke with the photothrombotic model, it's increasing the activation of astrocytes. Next, we confirm with the EBA1. It's the ionized calcium binding adapter molecule. So over here, it's expressed or activated these cells after the injury and help in the inflammation. So in the control sham animals, there is the very less the green color. However, it's the higher expression in the vehicle or the after photothrombotic stroke. So that's mean our model is representing that body weight loss, TTC standing infarction, hemoglobin, and activation of astrocytes and activation of microglia, all are the indicator of the brain death, brain cell death, and the inflammatory pathways. So for the long-term study, we perform the novel objective recognition test. So it's the test which the animals have the spontaneous tendency to explore the new things. So in this experiment, we use two similar object. So this is the novel object over here, but we use of the pink color object over here to familiarize the animal, and then we explore the next day how they explore the new or novel object. So over here you will see this is the novel object, and this is the circle is the familiar object. So in the control animals, they are spending more time towards the novel object. So this is the heat map. You can see blue color is very less intensity. The animal is walking over there and then the software is tracking, but you will see the red color is the high intensity of the animal. The presence of animal. So over in the group of photothrombotic stroke model, so they are spending more time in a corner. That means feeling sick or anxiety, or they have the short-term memory because they all these animals to explore the new things, but they are not exploring. So they are just feeling and they are showing the short-term memory. Both these, these parameters, we confirm with the recognition index and discrimination. Recognition means how they are recognizing and how much time they're spending. Discrimination means they are discriminating. They're spending more time towards the novel object. For the long-term memory impairment or the cognitive impairment, we perform the Morris water maze. It's the a pool. So we drop the animal, and there is the blue color of the inside the water is the hidden platform. So we drop the animal over here. So because the animals are the natural swimmer, so they can swim and then they can recognize the platform where is, because on the first day we keep the Q test and we put a stick on the platform so they can identify easily. And then after that we remove the tip test, and then we perform the final experiment. So in the disease condition, they are not able to find the platform. For this, we recorded this track plot. So you will see here. So they are spending more time in the target coordinates, and the vehicle is just performing. They are just swimming towards the peripheral. So we performed these tests for the 7 days. Day 1, we did not see any difference. Day 2, we did not see any difference, but day 3, it's a slight difference. Day 4, it's slight, and day 5, and at day 6 and 7, we found a significant difference. So and then we calculate the track plot, how much time they are spending in the target coordinates. So we saw that the vehicle with the photothrombotic, they're spending less time in the target coordinates. So overall, the summary of the model of robotic is significant with the respect and existing the method and future perspectives for this model. So one is numerous well-standardized models are developed in the rodents in order to closely mimic the human stroke pathophysiology and to develop new neuroprotective strategies. Second is this is a permanent focal ischemic model that constantly induces a stroke injury of the desired size in any cortical, subcortical site in a very reproducible and mainly invasive manner. So most important our point is over here that this model shows a high survival rate that reached 100%. So those are working in the field of stroke. I mean, you will people see the paper published on the PubMed. So this shows the mortality rate. But over here in our model, we modified it. So we found 100% survival rate. We did 40 animals and all animals survived till 31 days, while more extensive studies obtained 98%. So this is the advantage of our model. And furthermore, it is suitable for a number of experimental stroke studies, particularly for the evaluation of cortical plasticity in response to brain injury at both cellular and molecular level. For example, the model for the photothrombotic is the long lasting model because there's no mortality rate. But in case of the filament model, embolic stroke, and several hemorrhage, all these models shows the mortality rate is higher. So we cannot conduct the long-term studies. So only the people shows the 70 to 48, 24 hours, 24 hours, 48 hours, 72 hours. I mean, the maximum is the 77 days. So in our model, we are showing that the higher survival rate is the 100%. So we can check the neural plasticity and the effect of the ischemic stroke on the cognitive impairments and aging. We can see so many different things, and we can come out with a neuro strategic tragedy with a new compound of the drug or drug repurposing, whatever we can look at the drug model. So thank you.
Thank you so much for such an informative talk, Dr. Salman. Here, Dr. Charu ma'am is also your colleague and waiting to talk with you.
Hi ma'am, how are you?
So, dear participant, if you have any query, you can ask in between. Okay. So, our supervisor and Professor Rina Patnaik ma'am is also there. She has also a vast experience in the stroke field.
That's good.
Yeah. Okay. So, participant, you can unmute yourself and ask the query related to stroke, ischemia, ischemic models, thrombotic, embolic stroke, or he is the expert. Salman is the expert of all kind of stroke. TBI has also performed. So you can ask about the ischemic as well as hemorrhagic stroke also. Any question? Okay. Are you able to unmute or not? Charu ma'am, are you able to unmute?
Yeah, I'm
Okay,
able to.
okay.
Good morning,
It's
sir. Bhavna here.
moving.
Yes.
Yes, sir. Am I audible?
Yes, Bhavna.
Hanji. Sir, I have a question. What is the role of glutathione in ischemic cascading? Thank you.
Yeah, glutathione is okay. I will describe in this is the phase 1, phase 2 enzymes, glutathione and glutathione reductase, glutathione peroxidase, superoxide dismutase, catalase. These are the different enzymes that play a role in stroke as an antioxidant. So they just consume the free radicals and then convert into the the water, and they reduce the oxidative stress level. So in case of the stroke, it's goes down because it's the brain tissue damage. But when you uh, keep the treatment with the patient or in the animal model, so it's come up. So basically, it's important. The glutathione important role is in the phase 1, phase 2 enzyme reactions and play as a anti-oxidant role.
Okay, so thank you very much.
Okay, any other query? The session is open for the question answer. So you can ask your query. Do not hesitate.
Even though if you would like, you can write an email too, because right now we are going to publish this our research in the job protocol because they are very impressed. Why we are getting 100% survival rate, and it's the long-term study because few research groups in United States they are doing on the short-term studies. That's the best, advantage for us. So if you need any help and you would like to, introduce these model in your lab too, so we are happy to share all the information through the email.
Yes, the young MSc student who want to make their career in neuro protection or neuroscience field, there is this is the golden opportunity to discuss with the speakers.
Yeah.
Okay, any query?
I don't think so. They will be having any query because Dr. Salman was absolutely clear about everything.
Yes,
Wonderful.
yes.
Dr. Salman.
He
Thank you.
also discussed very basic things also.
Yes, yes, yes,
Yes.
yes.
Okay,
That
so
made
we
it.
are
That made it.
moving further, and we are inviting the Professor Rina Patnaik ma'am to kindly facilitate our speaker.
Thank you, ma'am.
Thank you very much, Mr. Mohammed Salman, first of all, for accepting the invitation and for very nice explained how the model is created and how you have very nicely explained the behavior and needs also and the cognitive function and all. So thank you very much. And I, on behalf of Galgotias University, please accept this moment.
Thank you, ma'am. Thank you so much. And thank you so much for the Galgotias University and Dr. Charu, ma'am, and all the participants, and especially Dr. Kumar.
Yeah.
Yeah.
Thank you.
It was
Thank you
nice
so
having
much.
you.
Thank you.
Okay, thank you once again, Dr. Salman, for accepting our invitation and joining this session for the enlightening talk and all the thing, the basic and all the advanced level of stroke and the innovative strategy of neuroprotection. So we are appreciating your research work also and wishing you all the best in your future.
Thank you so much.
Thank you so much. So we have another speaker with us, Dr. Sumedha. Uh, Sumedha, Dr. Sumedha, are you able to?
Yes, I'm able to hear you.
Okay.
Thank you.
Our next speaker for today is Dr. Sumedha Mukherjee. Currently, she is working as research associate in the University of Liège, Belgium. She has completed her PhD from Indian Institute of Technology, BHU, Varanasi. She worked on neuroprotective potential of endogenous hormones and vitaminoloids A in cerebral ischemia for her doctoral thesis. Her research focuses on involvement of neuroprotection cell death pathway in neonatal hypoxia ischemia, encephalopathy, and neuroprotective role of estetrol in hypoxic ischemia encephalopathy. Published more than 16 research articles in the field of neuroscience and neurodegeneration in reputed SCI index journals. She has published, filed 5 international patents, and completed several projects. Thank you so much, ma'am, for accepting our invitation. And over to you, ma'am.
Thank you, everybody. Am I clear and audible?
Yes, yes.
Okay, first of all, I would like to thank Galgotias University Institute Innovation Council for having me for the seminar today. A very good morning to you, ma'am. I'm very glad that Dr. Patnaik is here. She is my PhD supervisor. And thank you, Gaurav sir, for having me here. So I will start with my presentation. Okay, so going with the theme, I am presenting about exosomes, which are a novel potential tool which are currently used for neuroprotection. These are an emerging tool, emerging form of drug delivery, and I hope I will be able to shed some light on how it is used and what are its beneficial effects for combating the pathology that are the neurodegenerative diseases, and how these small molecules that are synthesized by our body can come handy in neuroprotection. So in this presentation, we will be covering the obstacles that are faced in the central nervous system drug delivery and use of exosomes as a therapeutic agent in 2 different stroke models. As Dr. Salman has already mentioned, that there are ischemic and hemorrhagic strokes. So we will cover how exosomes can be used as therapeutic agents in ischemic and hemorrhagic stroke models. And we will also see how it has been used in Alzheimer's disease. And also, as all delivery methods have some pros and some cons, we will finally cover how they have some advantages and disadvantages. So what are the major problems that are faced in drug delivery? There are lots of neurodegenerative diseases, and most potent of them, the stroke, the Alzheimer's, the Parkinson's, and other neurodegenerative diseases. What constitutes so much problem that till date we do not have a major therapeutic intervention for all these diseases? Why cannot we say that we have a drug that works very good on stroke? Why cannot we say we have a drug that works very good for Alzheimer's? Because technically we do not have many. For stroke, we only have one, the RTPA. So the basic reason why most of the systemically administered drugs in treatment of CNS diseases fail are the physiological barriers that are present in our body that actually exist to protect the brain. So their job is to protect the brain, which they do very well. And it's a very tough job for them. But the thing is, when they protect the brain, they also stop the molecules or drugs that we administer to enter the brain. So the first and most important of all these barriers is the blood-brain barrier. The second is the blood cerebrospinal fluid barrier. And the third is the blood tumor barrier, which often is present in case of a blood tumor or a brain cancer disease. So among all these 3 barriers, since the blood-brain barrier is the most important, let's talk a little bit about the blood-brain barrier. What is the blood-brain barrier? The blood-brain barrier is a physical and metabolic barrier which actually exists to protect the brain microenvironment from the external pathogens. This barrier is composed of several small blood capillaries and along with larger blood vessels, and they have specifically polarized endothelial cells which interact with each other, making junctions so that there are no toxins or pathogens present in the body can enter the brain and harm it. The endothelial cells make the tight junctions, and thus resist the entry of any kind of blood-borne substance in the brain. Beside the endothelial cells, the other major components are the astrocytes, which are like the main defense system of the brain, the neural cells, microglia, which is similar to the macrophages that work in our body, and some neurons, and the extracellular matrix components. And how does blood-brain barrier, because it exists to protect brain. So why is it a main issue for the drug delivery to the CNS? So the blood-brain barrier limits drug delivery by allowing only the lipophilic molecules or small molecules. So lipophilic molecules are those molecules which interact more with the water. So they only allow lipophilic molecules and small molecules. They will not allow any molecule with a higher molecular weight than 400 or 400 Daltons to enter the brain. And this is why nearly 98% of the small molecules that show some significance or some promises in the field of drug delivery, they do not finally survive the clinical trials, or they cannot be passed as a clinical therapeutics once they go from the lab to a larger scenario. So hence, due to the main opposition posed by the blood-brain barrier, nearly less than 10% of the therapeutic agents which are tried and tested in lab do not make finally into the clinical trials. So obviously, the scientists are not going to stay calm or take it as a obstacle that, okay, there is a blood-brain barrier, we cannot overcome it. So we come up with strategies. So the first, one of the most important strategies to overcome the obstacle of blood-brain barrier was the nanoparticle-mediated drug delivery. This is still a very popular technique for drug delivery in brain, and nanoparticles, since they're very small, they can be used to engulf the drug that we want to deliver into the brain, and thus they can make their way into the brain. The second is another innovative technique where we target the receptors that are present on the cells of the blood-brain barrier. So we can use different type of antibodies, different type of monoclonal antibodies, particularly directed towards the receptor of the brains and brain cells, and thus these antibodies can interact with the receptors present on the cells of the blood-brain barrier and enter the brain and convey their therapeutic potential. Next, since the blood-brain barrier allows the lipophilic molecules to pass, the surfaces of the drugs that we want to use, surfaces of the treatment drug or the therapeutics that we like, but they are not able to cross the blood-brain barrier, we can increase the lipophilicity of the drugs. So lipophilicity of the drug can be used by different kind of surface modulation, surface modification, thus making them able to cross the blood-brain barrier. Next, it's also a technique, but it's not very used in human trials, with the invasive techniques. That is to inject the drug directly into the brain so that it do not have to cross the blood-brain barrier. And all these techniques have been tried and tested for a long time, and we now have the new generation of drug delivery system, which is the exosomes. So why are we so interested about exosomes? What are exosomes? Exosomes are generally formed from the late endosomes. So before we start with exosomes, what are the endosomes? Endosomes are normally the intracellular sorting organelles. These endosomes, they regulate the trafficking of proteins and lipids and among the subcellular compartments and of the secretory and endocytic pathways, specifically along the plasma membrane, Golgi apparatus, and trans-Golgi network, and the vacuoles and the lysosomes. So what is actually the exosome? When the cell needs some components of it to transfer between the cell to cell, they create some small vacuoles, some small compartments, some small vessels from inside the cell, and the particle is transformed from one cell to another cell. So they are actually mediators of transcellular information. So exosomes are secreted directly from the cells. And so as we see, this is formation of an early endosome, where the cellular membrane here, the vesicles invaginate and forms a small compartment which carries the information like proteins or other signals that the cells want to transfer, which then mature into multivesicular body or MVB, where more small invaginations are formed, and finally they move as endosomes. And what happens then? So once the interluminal vesicles, the ILVs are formed, they are invaginated into more small vessels, which we finally consider as the endosomes, and they are released to the next cell, confirming the intracellular network pathway. So what is so special about endosomes? Exosomes. What is so special about exosomes? These exosomes are released from cells as signal mediators or for passing on information, passing on proteins. Since they're released from the cells they originate, they have lots of membrane receptors. They express lots of membrane proteins, which the cells recognize as their own. So when these exosomes are used as a drug delivery molecule, they can be not unrecognized by the cells. So when these exosomes reach the blood-brain barrier, the cells know them. They have already the receptors. So we don't have to technically modify them with receptors or anything. So to summarize, what are exosomes? Exosomes are tiny cellular vesicles which range from a size of 30 to 100 nanometer. They are found in normally all eukaryotic body fluids and mediate important physiological function. They have the properties that reflect the donor cells. That is, if an exosome is produced by the brain cells, they have the properties of the brain cells, like similar transmembrane proteins, similar sort of the proteins inside the cells. So they reflect the properties of the cells from where they originate. And they are produced in extremely abundant amounts because lots of all the cells in the body produce the exosomes. So how do we produce exosomes for therapeutics? Normally what is done is exosomes for therapeutic interventions are produced from the exosomes for therapeutic interventions are produced from the stem cells. So we take out the human stem cells, are retrieved from the body, they are cultured, and they are made to differentiate according to the pathology we want the exosomes to be the treatment for. So in this case, since we are talking about neuroprotection, I am focusing on the generation of dendritic cells. So we are differentiating the human stem cells into dendritic cells, which then release the exosomes and which can be finally delivered into the patient. So in this case, we are taking the cells from the patient's own body, using it as a treatment for their own. So the body do not think that these are some external compound. They do not think of them as a foreign element. So, but there remains the question that, okay, you are taking the cells out from the body and you are putting it back. It's very good. Well, we understand. But how do we put drugs inside it or whatever we want to put inside it? So exosomes can be used for loading of drugs using a very simple technique, which is incubation. So the desired cargo can, in this case, which is either the protein or the macromolecule or the drug that we want to use for neuroprotective effects, is incubated along with the exosomes that are produced by our cells of preference, in this case, the neurons. So they are incubated along with the exosomes and the exosomes take them up. They are incubated along with the exosomes and the cargo is uptaken by the exosomes and it's already inside. So if it is normally macromolecules or nucleic acids like miRNA or siRNA treatment, they can be easily incubated with the exosomes. But if it's proteins that have to be loaded into the exosomes, then it has to be performed with an optically reversible protein-protein interaction method. And why are the exosomes interesting therapeutic agents? Because we already have nanoparticles, we already have liposomes, we have lots of different methods for drug delivery. Why suddenly exosomes? Exosomes, the first point that I said, that they are already generating from our own body. So there is one problem, because it's generated from our own body, they can be cleared out of the body also very easily. But exosomes can be easily modified on the surface by adding the PEG, polyethylene glycol, which is a very simple technique called PEGylation, to modify the surface by combining it with PEG. It can be made to sustain in the body for a longer duration without it being cleared off the circulatory system. The next interesting thing about exosome is the surface proteins that it expresses. So it can express a whole array of membrane proteins depending on the cells they're originating from. So this is very interesting because this part can be exploited and is exploited for the specific targeted delivery. Since they are already expressing the transmembrane proteins, they are easily accepted by the target cells because they have the receptors. Next, and the most interesting thing that is interesting in the context of the neuroprotection, that exosomes can easily pass the blood-brain barrier, because with the membrane proteins they are expressing here, they bind with the receptors present in the blood-brain barrier, and they can enter the blood-brain barrier and thus go inside the brain to release the cargo they are holding, be it a nucleic acid, be it a protein, or be it anything. So they can be loaded with the macromolecules or drugs. They can be loaded with double-stranded DNAs or single-stranded RNA, depending on your choice of therapeutics, or even proteins, therapeutic proteins that we want to deliver into the brain for the treatment. So as I said, that we will be discussing how exosomes has been used for therapeutic studies in recent times, because this is a very new technique and it's still in its infancy. It's not even a very young technique. So I will discuss how exosomes has been used in an ischemic stroke model. So as Dr. Salman previously said, that there are lots of different stroke models that are used. In this particular study, the stroke model that has been used is the middle carotid artery occlusion, where the middle carotid artery of the mice was occluded for 40 minutes, followed by a reperfusion. And after 1 hour of reperfusion, the mice were administered with 10 microliter of exosomes derived from the mesenchymal stem cells, and for the treatment continued for 7 days, after which the mice were sacrificed to check the effect of the therapeutic effect of the exosomes. So before we check what has been done in the literature, I would like to talk about what are the main attributes of ischemic stroke, so it's easy for our listeners to relate. So the first main criteria is the cerebral infarction, where we see the ischemic core and penumbra, and it is detected by the TTC staining, and cerebral edema, where there is a high water content in the brain, there is an increase in the brain water content, and there is the BBB damage. So what do we see in this model? We see that in the middle cerebral artery stroke model, and the mesenchymal stem cell derived exosomes, which has been loaded with curcumin, show a very good effect in reducing the cerebral infarction. So normally the sham is the animal that has been operated, but there has been no surgery performed for induction of cerebral ischemic stroke. So here we see that normal in sham, there is no effect of the exosomes along with the curcumin, but in the ischemia reperfusion model of ischemic stroke, we see the infarction here, the white part. And we see here also that how it is reduced by treatment with the mesenchymal stem cell derived exosomes along with curcumin. After the infrag volume is confirmed that they have a good effect, good therapeutic effect in reducing the cerebral infarction, let's check the brain water content. We see similar effects here also, where we see the brain water content is very significantly reduced while with the treatment of the exosome loaded with curcumin. After the cerebral edema, there is another important factor in cerebral ischemia pathophysiology, which is generation of the reactive oxygen species, which causes which actually causes the downstream of the ischemic pathophysiology cascade and causes the cell damage and lipid peroxidation of the membrane, causing neuronal cell death. So here the ROS activity of the brain cells were checked along with the treatment of MESC, that is the mesenchymal stem cells derived derived exosomes in the ischemia reperfusion model, and there was a reduction in the production of the ROS. After this, it was checked how the exosomes loaded with curcumin affect the blood-brain barrier. So here we have an immunofluorescence study which looks at the proteins that are expressed at the blood-brain barrier. So the proteins that are looked here is the intercellular adhesion molecule 1, or the ICAM. As you can see, it's the green staining here, and the VE cadherin, which is in the red staining. So before we look at the images and we are like, okay, we see only green and red, what are ICAM and what are VE cadherin? The ICAM is a molecule known as intercellular adhesion molecule 1. So how is this implicated? Higher level of ICAM1 is expressed in most of the pathophysiologies, where it rearranges, when it's overexpressed, the ICAM, it rearranges the actin cytoskeleton to cause cell elongation and thus alter the junctional integrity. So the tight junction that is formed between the cells in the blood-brain barrier is altered when there is an increase in the ICAM production. And why is there an increase in ICAM production? In this case, it's the ischemic pathophysiology, it's the ischemic insult that causes the degeneration of the blood-brain barrier, which can be understood when we see there is an increase in the ICAM production. So as we see here, normally in the sham, which is the control animal, we see very less of the green fluorescence. And it's same in case of the sham treated with the exosome-loaded curcumins. But in the ischemia reperfusion model here, if we check that there is a high fluorescence, high green fluorescence that can be observed here, representing that there is a overexpression of the ICAM protein. And when it is quantified, it can be seen that there is an increase also in the ICAM protein. Next comes the VE cadherin proteins. So what are VE cadherin proteins? We now know that ICAM are overexpressed, but what does VE cadherin has to do with it? So VE cadherin is another junction protein which is found in between the endothelial cells. So VE cadherin normally is a symbol of tight junction formation between the endothelial cells. So when there is a depletion in the expression of the VE cadherin, it will mean that the tight junction has been disrupted. So we can see here also in the red staining that in the sham animals, the VE cadherin is highly present. There is a red fluorescence similar to the sham with curcumin with exosomes. But if we look at the ischemia reperfusion model, there is almost nothing. We do not see at all the staining of the VE cadherin here. The red staining is almost depleted. But again, in the therapeutic treatment with exosomes, we see the VE cadherin proteins are expressed. So when the images for histoimmunofluorescence are quantified, we can see that in ischemia reperfusion model, there is an increase in the ICAM1 protein expression and a reduction in the VE cadherin 1 expression in the ischemic reperfusion or the stroke model, but with the treatment of MSC-derived exosomes loaded with curcumin, the ICAM levels are decreased and the V-cadherin levels are increased again, which is highly significant when compared to the ischemia reperfusion models. As we see here, it is almost less than 0.001. So this expression, the change in regulation of V-cadherin and ICAM expression is highly significant, and it signifies that the exosome therapy here has successfully ameliorated the blood-brain barrier disruption. But to further understand how the blood-brain barrier is working or how the exosome can be beneficial, so 2 proteins is okay, but 4 is better. So we have here 2 more proteins whose expression is checked for the check the function of the blood-brain barrier, how if it is intact or not. So these 2 proteins here are claudin 5 and occludin. So why is claudin 5 significant? Claudin 5 is one of the most abundant tight junction protein present in the blood-brain barrier. And its dysfunction has been highly reported that dysfunction of claudin 5 is highly significant in any kind of neurodegeneration disease. In any kind of neurodegenerative disorder, when the blood-brain barrier is affected, claudin 5 is the expression of claudin 5 is decreased because the membrane disintegrates and the protein is not expressed again. And similar with the occludin, it is also a tight junction protein, and it is shown that rather than the assembling the cells to form the tight junction in the blood-brain barrier, the occludin protein is very important to maintain the stability of the tight junction in the blood-brain barrier, and also to maintain the stability of the blood-brain barrier. So depletion of occludin protein will mean that the blood-brain barrier has been compromised. So here also we see in the control animals, the occludin 5 and occludin and the claudin 5 are highly expressed. It is a normal expression of the proteins in the sham group, but in the ischemia reperfusion group, there is no expression or less expression of the claudin 5 or the occludin protein. And with the treatment of the exosomes, curcumin-loaded exosomes, the claudin 5 and occludin are again expressed. And this also, when quantified and statistically analyzed, show that it has a significance of p-value less than 0.001, making it highly significant. Making it highly
Dear
significant.
participant, please mute yourself. We will give you the sufficient time for the discussion.
No, if anybody has a question, it's fine.
Okay. In between the
Is there a
session, if
query?
you have any question. Okay.
Okay, I think I should continue. So
you can continue.
Thank you. So in this case, what we see here is that with the treatment with the exosomes loaded with curcumin, we have the increase in the expression of both claudin 5 and occludin, signifying that these exosomes show a protective effect in maintaining the integrity of the blood-brain barrier. So now we have seen the effect of the exosomes in ischemic stroke model. The next model that we will look at is a hemorrhagic stroke model, where blood infusion is injected directly into the stratum of the rat to induce hemorrhagic stroke. And after 24 hours, the mice are injected with tail vein injection of 100 microgram of protein of mesenchymal cells, stem cells derived exosomes. And this is only the exosomes. This is pure exosomes derived from the stem cells, and they do not have any kind of drug loaded into them. So we will see the role of naive exosomes or native exosomes in neuroprotective capacity. So in this study, two different studies were done, but they are mostly depending on the behavioral tests. So a Morris-Water maze test was done to check for the effectivity of the exosome in improvement of spatial memory. And it was observed that for a time duration of 28 days, as is done in case of the behavioral studies, where it is a longer time duration to check how it improves. And it is seen that with the increase of time, the significance of the study increases. Like, it takes them less time. Morris-Water maze test is based on the time mice takes to find the platform between a water maze. So with time, it is observed that the mice takes less time to find the position it has to search for. So thus, it can be concluded that the exosome treatment improves the spatial learning in the mice who have been subjected to ICH or intracerebral hemorrhage stroke. The other study that was done was the MNSS, the Neuro Severity Score, and which is done using several different kind of behavioral tests like Rotarod or order test. And here also there was a significant improvement at the end of 25 days. So with two different stroke models, we have seen now that exosomes can modulate the neuroprotection, they can improve the blood-brain barrier, they can confer neuroprotective ability, they have the neuroprotective ability by reducing the ROS, they can improve the cerebral infarction, and they can also improve the memory. So after the stroke models, we will move on to another interesting neurodegenerative disease, which is the Alzheimer's disease, which is one of the most occurring neurodegenerative disease and which have again no therapeutics to study. And here the therapeutic potential of exosomes are checked in the Alzheimer's disease model. And for this model, we have thankfully lots of transgenic mice. And here the study uses APP-PS1 transgenic mice. So what is an APP-PS1 double transgenic mice? So this APP-PS1 double transgenic mice expresses a chimeric mouse human amyloid precursor protein, that is the APP, which is the amyloid precursor protein, which is derived from both, which has the gene from both mouse and from the human. So it produces a mutant human amyloid precursor protein and also a mutant human gene, that is PS1 or presenilin 1. So it has two different transgenes in it, and this transgenic mice is a very well-known model to establish the Alzheimer's disease. And these both mutations, APP and PS1, are implicated with the onset of early onset Alzheimer's disease. Just as we looked in the markers for stroke, we have to first check what are the markers or attributes that we have to check when we are looking at an Alzheimer's disease to know how they are modulated or how they are regulated or how they have been benefited. So first marker for Alzheimer's disease is the formation of the beta amyloid plaques, which are formed in between the neurons, which hinder the signal transmission between the neuronal cells. The second is downregulation of the amyloid beta degrading enzymes. So if the plaques are formed, there cannot be any neuronal signal transmission. And if the plaques are degraded, it is far better because the cells regain normal they can reactivate their normal neuronal transmission system. But in this case, when there is a prevalence of the Alzheimer's disease, the enzymes that can break this little plaques, they are downregulated. And the third is the activation of microglia. The microglial cells that are present in abundance in the brain are highly activated during the Alzheimer's disease and contributes to the pathophysiology. So As we look for the first slide, and our first marker was the formation of the amyloid beta plaques. So what do we do to check for the amyloid beta plaques? Just as we use the TTC staining for the measurement of the infraction volume in stroke, we use a thioflavin S staining in detecting amyloid beta deposition. Thioflavin S is a fluorescent marker of amyloid beta protein formation, amyloid beta plaque formation. And here with the green fluorescent dots that we see here are the accumulation of the amyloid beta protein that form the amyloid beta plaques during the Alzheimer's progress, because Alzheimer's is not progress in a single day. With the progress of Alzheimer's, so we see the progress in the formation of the plaques. So when there is a treatment, and this we check in both hippocampus, where there is a high formation of the plaques, and also the cortical region of the cells. So this is the hippocampal region and the cortical region. And when we treat the mice with the exosomes, and these are naive exosomes without any drugs, so the exosomes show the effect, and we can see clearly that there is no such accumulation of the amyloid beta plaques as was observed in the control model. The plaque level is highly reduced in the hippocampus as well as the cortex. And when it's quantified, it shows that it is significant with a p-value of less than 0.05. So after the formation of the amyloid plaques, we look for the next marker, that is the regulation of the enzymes that break the amyloid beta plaques. So what are the enzymes most important for degrading of the amyloid beta plaques? One is NEP, which is the neprilysin, and one is the IDE. IDE is the insulin degrading enzyme. The IDE or insulin degrading enzyme degrades the amyloid beta plaques, and both proteases are involved in the degradation of the amyloid beta, and they are most of the time downregulated during the late onset Alzheimer's disease. And here we have a Western blot result where we check the expression of both of these proteins and compare the control group and the exosome treated group. And the exosome treated group, and we see in the control group, that is the mice with Alzheimer's disease, have a lower expression of both neprilysin and insulin degrading enzyme IDE, which is overexpressed when the mice are treated with the exosomes. So when these enzymes are overregulated or they are upregulated, they will be able to break down the amyloid beta plaques and thus help the pathophysiology to not prolong or progress further. After the plaque formation and the regulation of the amyloid degrading protein expression, we look for the expression of the microglia. The microglia cells are stained with IBA1 protein antibody, because IBA1 is one of the proteins that is expressed on the membrane surface of the microglia. And as we can see here in cortex and hippocampus, in the control mice, that is the Alzheimer model of mice, there is a high expression of microglial activation. So when microglia is activated, they produce the protein IBA1 on the surface, and it can be seen with the red staining that we see here. And when we compare with this expression of the same protein in the MSC exosome treated mice, we see that the expression of activated microglia is reduced. So there is a reduction in the microglial activation in the brain regions of cortex and hippocampus in the exosome treated Alzheimer mice. So what will this prove? That there is the exosomes reduces the inflammatory activation of microglia in vivo. And after microglial activation, what comes after that? Microglia is like the macrophages of the cells. So normally in bodies, the macrophages work as the immunomodulatory cells. They are the guards of our cell. And how do they do it? They release the cytokines. So there are some cytokines that causes the inflammation, and there are some cytokines which controls the inflammation. So in this study, two different type of cytokines were studied. So the cytokines that promotes inflammation are the pro-inflammatory cytokines, and the cytokines that reduces the inflammation are the anti-inflammatory cytokines. So what do we see here? We have interleukin-1 beta and TNF alpha and TGF beta and interleukin 10. Of these four cytokines, interleukin-1 beta and TNF alpha, these both are pro-inflammatory cytokines. And TGF beta and interleukin 10, they are the anti-inflammatory cytokines. So we see the amyloid beta model is marked in black. So in the amyloid beta model, when the expressions were checked using an ELISA study from the cells of, and this was not checked in the in vivo cells, this were checked in the cell culture medium. It is an in vitro study from the same paper where they have checked in the microglial cells the effect of exosomes on the microglial cells and how the microglial cells are activated or modulated. And we see here that the cells produce an increased amount of pro-inflammatory cytokines, that is interleukin 10 and TNF alpha, which is reduced by the treatment with the exosomes, and there is a reduction in the production of the TGF beta and IL-10, which are the anti-inflammatory cytokines in the Alzheimer's disease model. And when they are treated with exosomes, there is an increase in the production of the anti-inflammatory cytokines TGF beta and interleukin 10. So thus, from these three different models of neurodegenerative diseases, we can say and summarize some of the beneficial aspects of the exosomes for combating the pathophysiology of neurodegenerative diseases. So what do we see first? From the stroke models, we see that the exosomes are able to confer neuroprotection by regulating the cerebral infarction, by reducing the cerebral edema, by modulating the proteins expressed in the blood-brain barrier junctions. So they are very good agents to confer neuroprotection in several neurodegenerative disorders. And we saw in the ICH model, the intracerebral hemorrhage stroke model, that treatment with exosomes can also improve the behavioral changes that are often associated with the stroke pathophysiology. Also, we saw that they reduce the inflammation and modulate the immune system in the Alzheimer model, because they reduce the activation of microglial cells, they reduce the pro-inflammatory cytokines, they increase the expression of the anti-inflammatory cytokines. So in thus, the exosomes also can function as immunomodulators and can reduce the inflammation that is correlated with the neurodegenerative disease onset. And the most important thing that we have observed here is that exosomes also reduce the plaque formation in Alzheimer's cells, which is a very important point because the amyloid beta plaque formation is a very significant attribute of the Alzheimer's disease, which is the main target of lots of studies. So it is one of the main and basic targets sort of that we have to reduce the production of the amyloid beta plaques to improve the condition of a Alzheimer's disease patient. Besides the studies that we had here, there are some other attributes of exosomes that has been reported in neurogenesis, in neurodegeneration, are that they induce neurogenesis, that is formation of new nerve cells. They can also induce formation of the synapses, new synapses, and they can also affect the white matter remodeling in the brain. But like all studies, all studies have their pros and their cons. But luckily for us, the exosomes has more pros than it has cons. So the main pros of the exosome therapy is that it can be used for a wide range of cargoes, that is the therapeutic molecules for neuroprotection. Like it can be used for nucleic acids, it can be used for protein delivery, it can be used for the drug delivery, like big macromolecular drugs. And one of the most important thing is, secondly, is that they are biocompatible. So there is no chance of the toxicity. So they have a very low toxicity. Since they're revived from the cells themselves, they're highly specific to the cells that we want them to be delivered to and not go to other cells. Like if we want to have a targeted delivery to brain, it is assured because they have the receptors or the membrane proteins from brain. They are not delivered to the liver. So they are highly specific to their target cells. They have a high cell permeability because of the proteins they express. And obviously, since they are very much biocompatible, they have low toxicity, and also they're very low immunogenic. So why is this part also important for exosome therapy? Because whenever we have some of the drug therapy or antibody therapy, there is always a chance that our body recognizes it as a foreign substance and it triggers its own immune system, which clears out the drug or the effects and can also harm the body itself. The immunogenicity of any drug can harm the pathophysiology, can harm the physiological systems of our own body. So it is a very good benefit for the exosomes that they show very low immunogenicity. And after the pros comes the cons, which are not so much, but since the idea behind any therapy is to take it to the next level, to bring it from lab to the market. So we need further clinical trials. So the exosome purification and isolation methods are only limited to production in labs for the scientific studies, but there are not yet optimized scalable methods for which has been produced for exosomes, which we hope will be done in near future. And there is also required the optimization of the drug loading approaches for in a larger scale. When they're produced in a larger scale, it needs to have efficient and optimized drug loading ability for the exosomes. And there is also another thing that we have to keep in mind when we are talking about a therapeutic application in humans. That is, we are talking about on-the-shelf drug, not only in the lab. So from the lab benches to the shelves of the market. So they have to be stored. They have to have a storage ability. So we cannot just make them and just use them on same day. They have to have a long time storage. So this is also a factor which remains an issue with the exosome therapy, that there are still not many methods which can protect them, or they can be stored off shelf for a long time. So thus, I would like to conclude that exosome therapy is still in its infancy, but in its infancy, it holds a very good and wide promise to become one of the very interesting and effective therapeutic technique. It has already proved so in cancer because it is already in clinical trials for cancers. And we really hope that in coming days, we will see the same for the neurodegenerative diseases, and exosomes will be an answer to our never finding drugs for neurodegenerative disorders. So thank you all, and I'm open to questions.
Thank you so much, Dr. Sumedha. You have discussed a lot about the exosome, their pros and cons. So scientist is currently working to enhance the stability and loading capacity of the exosome material. The students who are interested in nanoparticle and nanotechnology, as well as in neuroprotection, who can ask your query from the speaker. If you have any query, you can unmute yourself.
Excuse me.
Yes.
Yes, sir. Good morning, ma'am.
Hey.
Bhavna here. Hi. Ma'am, my question is, you have discussed about mesenchymal cells from
Yes.
animal, right? Like
Yes.
if I load a drug in a nanoparticle and I have to do its in vitro analysis for the neuroprotection
Okay.
purposes to
Okay.
analyze its neuroprotective impact. So what kind of cell lines I should use, and are they commercially available? I can acquire them commercially, or we have to get it in the lab only. Thank you.
No. Okay, Bhavna. So you are trying to do an in vitro assay with the neuronal cells, right? For neuroprotection.
Yes,
That is the
ma'am.
basic idea. Okay. So do you want human cell lines or you want mouse cell lines?
Mouse cell lines.
Okay.
Would
So the
which one sorry to interrupt, which one would
no,
be
no,
better?
please continue.
Yes, which one would be better, whether I should analyze the impact on the human cell line or the rat cell line,
Okay,
this
so
mouse cell line?
for this question, my answer would be that to use both the cell lines. Why why do you want to use one? So use both the cell lines. So there are very easily available, commercially available cell lines. I think you can get it from the ATCC if you want, or if not, we have a cell archive itself in in Pune, I think. And I don't remember the repository right now, but we have a cell archive in Pune. So there are two cell lines that you can use. The basic idea for using cell lines for in vitro neuroprotection purpose, it's very good to use cell lines that are neuroblastoma. Okay, so normally if you want to check your effect of your drug, so you need neurons, right? But if you harvest your neurons from a rat or a mice, this is also a doable thing, that you can harvest primary neurons, but they won't last longer. Like they will die within 1 month, maximum 20 days. So they will not survive more than 20-25 days. So if you want to continue a cell culture, so it would be good idea to get the neuroblastoma cell lines. So I can recommend two cell lines. So for humans, for human cell line, you can use the SHSY5Y. I don't know if I'm clear. I will repeat, it's
Yes, ma'am.
SHSY5Y. So this is a human neuroblastoma cell line, and it can be differentiated using retinoic acid. So you culture the cells. Since it's a neuroblastoma cell line, it will grow fast, but not very fast actually. It compared to normal cancer cell lines, it takes about 3 to 4 days to be confluence. So once it reaches 70% of confluency, you can add retinoic acid. There are protocols available online. So this is an established protocol. You can use a retinoic acid treatment to differentiate the neuroblastoma cell lines to fully matured neurons. So you will have your neurons. It's a bit time consuming. It will take you like 17 to 18 days to have fully grown neurons, but you will have your neurons. And another cell line that works, this is the human cell line. And the other cell line that is workable and it's very good and it's been used is the Neuro2A. So this is a mice cell line. So you can use both of this cell line. Neuro2A grows very fast and you don't have to wait 5 days. So it is like confluent in 3 days. So you can use Neuro2A and SHSY5Y for your in vitro studies. And you will find lots of literature to back up your research, because there are lots of studies, because you need to have references to have your study substantiated. And these two cell lines are very widely used for in vitro studies. So you can use both of them or any of them. I suggest use both of them.
Thank you very much, ma'am.
It's not a problem.
Thank you. Thank you, Bhavna. Any other query? Okay. So you have time for 2 minutes. So you can unmute yourself and make the session interactive. The speaker is open to address your all the query related to the neuroprotection and drug delivery strategy. This is the innovative strategy the scientist is using nowadays to not only address the neuroprotection, it can be also used in cancer also.
Yes, the drug delivery therapies are not restricted to one particular disease. So
Yes.
it can be if you are working in cancer or you are working in like other pathologies, drug delivery techniques are same. Like the preparation is the same, but you modify it according to your pathologies.
Okay. I think there is no query. So I would like to request the Professor Rana Patnaik ma'am to facilitate our speaker. Rana Patnaik ma'am. Your voice is breaking.
Ma'am, it's breaking.
Action family members, Sumedha, all three of you were very good. Yeah, so I'm so happy.
Thank you, ma'am.
Excuse me, sir. Hello, sir.
Yes, yes.
Hello.
Yes, yes.
Sir,
Yes,
myself
please.
Pallavi. And,
Okay.
and sir, I have asked one question from ma'am. Ma'am, can you please tell me a little bit about anesthetic neuroprotection?
You want to know about anesthetic neuroprotection?
Yes, ma'am. Ma'am, recently I have read a one article, review article, and there it is mentioned that anesthetic neuroprotection is regarding for, I mean, pharmacological properties. So, what is the correlation between them?
So what kind of, you have to be a little bit more specific, Pallavi. So what is the anesthesia that you want to use? Because anesthesia can also be fatal.
Okay, ma'am. Ma'am, basically, that article define the anesthetic process. It release the increase the tolerance of brain and ischemic insult, that is ischemic stroke in heart.
Yes, actually, what can happen is anesthesia, it actually anything is dose dependent. So you have to think about the dose here. Since you are speaking from a pharmacological point of view, so anesthetics, normal anesthetics, like a chloroform or I don't know what you have been using here or what the article suggests. It would have been good if you remember the. Do you have the paper with you?
I think she has read go through with some article like pentabarbital, sodium, as well
Okay,
as
okay.
ketamine,
So,
that
ketamine, yes,
are
yes, but
neuroprotective.
okay. So, so Pallavi, I understand the point. So, ketamine is actually a neuromodulator. So, if we talk about ketamine here, so ketamine is actually a neuromodulator. So, it modulates
And
the
also after treatment with ketamine, the body temperature goes down. Means
exactly.
hypothermia
Yes.
is created, and
Yeah.
hypothermia is always neuroprotective.
Always neuroprotective. Just
Yes.
like Gaurav sir said, that ketamine always lowers the body temperature, so it will be neuroprotective. But when you are using a drug, just like we were discussing with exosomes or nanoparticles or liposomes, so whenever you are using any kind of drug, you have to think also about the other side effects. Like ketamine obviously reduces the hypothermia, but ketamine also can modulate the neuronal transmission, the synaptic transmission of the signal transmission between the synapses. And trust me, that will not be very good all the time because ketamine, when not regulated or there is a problem with the dose, it can be a hallucinogen.
Okay, ma'am. Okay.
So,
Actually, sorry, Sumedha. Sorry.
yeah.
Yes.
Actually, if the hypothermia means it goes beyond the threshold level, then it is
Then
fatal to the animal and it may die. So
exactly.
you are
That is what
right.
I was
Like,
also going to say here because in neonatal HIE,
yeah,
hypothermia is one of the major treatments because there is also no drugs for HIE. So
yeah.
therapeutic hypothermia is used for HIE babies, neonatal babies. And when and in lots of cases, it has been seen that When hypothermia goes beyond the point, it has a high reperfusion of blood, which can even lead to the death, fatal to the baby. So it actually depends on dose. It actually depends on the time. It has lots of other parameters that has to be considered when you are administering anesthesia or any kind of therapeutic treatment.
Okay, ma'am.
And
Oh,
and
Yes,
nowadays,
Salman sir has some questions.
and nowadays, Sumedha, like ketamine is not used that much, because now several others anesthetics have been
Yes,
proposed. So because
yes,
ketamine interferes with the other neuronal aspects of the
yes.
organism, so that's
Yes,
why.
that is what I was trying to say to Pallavi, that
Yeah.
it will modulate the synaptic regulation of neurotransmission. And
Yes. Yes.
normally nowadays we are using the isoflurane is the best, I think,
Yeah.
bullet used. So we really do not use ketamine anymore.
Exactly. Exactly.
Yeah,
ma'am. Okay,
chloroform
ma'am.
has banned, restricted the ether. Previously, ether was
Ether,
also
yes.
used, so that is also restricted by Animal Ethical Committee. Pentabarbital sodium is also restricted.
Yes.
Yes,
Ketamine is still not restricted, but ketamine is not very widely used.
Widely used for, actually, in neuroscience, ketamine is having different effects on the physiology and behavior of the animal, so you do not know
Yes,
whether
yes.
it is the drug which is causing that
Yes,
effect
exactly,
or.
exactly.
Yes.
Yes,
So
I completely agree. That is what I was trying to explain, that even if it's not controlled, it can be a very potent hallucinogen.
exactly.
okay. Any other query from the participant? I think there is no query. So thank you once again, Dr.
Thank
Sumedha,
you.
to
Thank you, Gaurav sir, for inviting
to
me.
speak here from your busy schedule. We have our another speaker has also joined, Dr. Dhanananajay Kumar, sir, and the Shivangi. Over to you.
Yes, sir. Now for our next speaker, we have Dr. Dhananjay Kumar. He is currently working as research associate in Lab of Integrative Physiology, Migal Galilee Research Institute, Kriyat, Shmon, Israel. He has received his PhD from Department of Zoology, Banaras, Hindu University, Varanasi. He worked as CSRI, PDF, and SERB Young Scientist Fellow in Department of Pharmaceutical Engineering and Technology, IIT BHU Varanasi. Also worked as junior scientist in Grow Laboratory, Narayanan Netralaya Foundation, Bangalore, and as SRF in Department of Zoology, BHU Varanasi. Qualified NEET and GATE examinations, received Best Poster Award in 12th National Symposium on Chronobiology organized by Department of Zoology, Kurukshetra University, Kurukshetra, Haryana. He was also selected among top best 4 posters in international school, 5th UCLOC and 19th European PhD School of Chronobiology held in Jawahar Nehru Center for Advanced Scientific Research, Bangalore. He has published more than 30 research articles in the field of neuroscience in reputed SCI Index journal. Thank you so much, sir, for attending our event. Over to you, sir.
Dr. Gaurav, can you see my screen?
Yes, sir. Previously, you were presented, but now your screen is not visible.
Okay, so just a minute. So entire screen. So can you see the screen, or my?
Yes, sir. Yes, sir. Yes, sir. Now your screen is visible, so you can now open your presentation.
So can you
Yes,
see
sir.
the
It is
presentation?
visible. Yes, sir. It is visible.
So, dear colleagues, friends, and Ranjana ma'am, it's a great opportunity for me to present before you my research topic. And first of all, I would like to wish all of you Vande Mataram, because I'm far from India and I'm really missing India. So now the topic of my research is basically I have been working as Shivangi has introduced me that I have worked on circadian rhythm. So my till date I have been working in circadian rhythm, and now I'm working on the metabolism and circadian disruption here in Israel. I'm going to present my some of my postdoc work which I did in IIT BHU. So the topic of my research is circadian disruption due to chronic jet lag shift work, which leads to cognitive deficit. And I have used the model mice. So effect of circadian disruption as a result of shift work or jet lag, which causes cognitive deficit in mice. So this is the topic of my research. So first of all, some of the basic terminologies I would like to introduce. So what does circadian means? So the word circadian means that a phenomena which occurs in a day, that is within a 24-hour time period. So such phenomena are called circadian. There are other rhythmic phenomena which are infraradian, that is more than 24-hour, like the human menstrual cycle, or the distress or estrous cycle in what do you call the rodents. Then the ultradian cycle, which we see in the rapid eye movement, which lasts for 90 minutes. So ultradian means less than 24-hour. Example, another example is the growth hormone cycle, which lasts for every 3 hours. So it's a pulsatile. After every 3 hours, it reaches its peak and then it troughs. Another type of rhythm, it is called the circadial. So the word circadial means which rhythm which occurs after every 12.4 hours. Then the circalunar, which a rhythmic phenomena which occurs after every 28 days. You could see the moon, like after every 28 days you could see the full moon, and then it starts shortening. Then the circa annual, a rhythm which occurs or which takes 12 months or so. Example, the flowering plant in some of the trees, like in mango, if you see, the flowering will occur once in a year. So you will get the fruit only once in a year and not throughout the year. So these are some of the definitions of the rhythm. So basically here I'm focusing about the circadian rhythm because my research involves circadian. So first of all, where the circadian rhythm occurs. So the circadian rhythm, the center of the circadian rhythm is located in the hypothalamus, the floor of the hypothalamus, which is called the suprachiasmatic nuclei, or the SCN. It is a biological clock, or you can say as in Hindi as a Javik Ghadi, which is located in our brain. And it consists of a paired lobe of network of nucleus, which is separated by the third ventricle, and the connection of these lobe, they are both interconnected, and they are connected. I'll show you in the next figure. They are connected to the another center, or this sensory center in the our brain, that is the eye, which sends the light and then transfer that light information from the retina to these centers. And these centers are differentiated into the core, and the outermost is known as the shell. And the optic chiasma is the point where the nerve transmission from the retina comes and deliver the information to this what you call the suprachiasmatic nuclei. So basically in the suprachiasmatic nuclei, so there are millions of cells, and these cells are regulated. These cells causes the means production of different proteins, which regulate the expression of different genes, which further regulates the expression of different proteins, and it is a cyclic phenomena. So this interaction between the protein and the gene regulate the various physiological functions, like our sleep wake cycle, our digestion, our what you call other physiological and metabolic processes like the hormonal regulation. So the molecular machinery of the circadian clock involves basically two proteins, that is the clock and the BMAL1 protein. These two proteins in the cytoplasm of the suprachiasmatic nuclei or SCN cells, they dimerizes to form the heterodimer, and then they translocate into the nucleus where they bind to the promoter region of some of the clock genes, which and causes the upregulation of those clock genes like the period 1, that is the per gene, then the cryptochrome gene, that is the cry genes, and several other clock control genes. So after its regulation, these period genes and clock genes, they translocate to the cytoplasm where they get converted into different proteins. This phenomena occurs gradually, and what happens, like from morning till evening, the concentration of period and the cry protein keeps on building, and in the night when the amount of period and the cry protein reaches a maximum level, it goes and bind back to the clock and BMAL1, and it regulates its when it binds to the clock and BMAL. What happens, it causes the dissociation of the clock and BMAL from the promoter region of the genes, and which leads to stoppage of the production of PER1 and PER2 proteins. That means the clock and BMAL1 act as a positive feedback loop, and the period protein and the cry protein dimeric form, it acts as a negative feedback loop. So in this way, when the concentration of one gene is low, then the clock and BMAL1, they bind and they cause the upregulation of period 1 and period 2 protein. So in this way, the cyclic phenomena occurs in 24-hour day-night cycle, and these causes the regulation of various physiological and metabolic processes in our body. So some of the cues which are regulated by the or some of the cues which regulate or which affects the circadian clock, which is also known as the master clock, are the light. So light, when passes through the retina, so retina have different photoreceptors. These photoreceptors are known as the RGC, retinal ganglionic cells, and the innervation from each retinal ganglionic cells are joined together into a network of neurons, and it transfers the information. It transfers the information from the.
Okay. So, dear participant, please be muted during the presentation. If you have a query, we will open the question and answer session. Okay. Sorry, sir, for the disturbance. You can continue.
No, it's okay. No problem. If they want, they can ask. I can.
Okay.
So,
okay.
So what happens if there is any means the information from the eye is transferred to the master circadian clock, and these master circadian clock, which is located in the suprachiasmatic nuclei in the brain. So those these clocks further regulate the other physiological and behavioral processes. So out of several cues or the stimulus, light is considered to be the most important and most powerful cue. So that's why nowadays you keep on hearing that you should not watch mobile or laptop or TV in the late night. Why? Because during the late night, there is a hormone known as melatonin, which is produced by the pineal gland, and this melatonin is at its peak or reaches its peak in the late night time. Around it starts building from 10 and reaches its peak around 1 o'clock in the night. So if the light is being transferred from the eye to the SCN, what happens? The SCN sends signals to the pineal gland, and then it stops this secretion of the melatonin. And melatonin is such a important hormone which has pleiotropic effect. It regulates many physiological and biological functions like reproduction, sleep wake cycle, and several others, digestion. So when the melatonin is suppressed due to untimed exposure of light, various disturbances in the physiological, metabolic, and biochemical processes occurs, and that may lead to various kind of disorders in our body. Apart from the light, another cue or the stimulus is food, which is also one of the key important thing nowadays. A lot of research is going on the effect of different kind of diet, and at how many times we should take diet, and if we restrict our diet or do fasting, which in our tradition, Indian tradition, very common, doing fasting. So nowadays in the European or in the Western world, there is booming or a lot of research is going on about the fasting, keeping fast, and how it is regulating our uh, causing several beneficial effect to our body. Apart from the food, another stimuli or the cue which control or may regulate the circadian clock is the sound, and another one is the drug. So these are all several types of cues or the stimulus which may regulate the suprachiasmatic nuclei, which is the master clock or the Javik ghadi in our body or in our brain. So you see that from the light from the when the light enters the eye and through the retinohypothalamic tract, it goes to the suprachiasmatic nuclei, and from the suprachiasmatic nuclei, several signals are processed and passed to different centers of the brain as well as to different parts of the body. So you could see that the SCN is being directly or indirectly affected by the pineal gland, which produces the hormone melatonin. Then the SCN also regulate the sleep wake cycle through another pathway, and you could see that the SCN is also affecting the wakefulness and the feeding behavior. So, and the SCN is also being affected by the raphe nucleus and intergeniculate leaflet. These are all the centers which are present in the brain. So I don't want to go in such a detail, but I wanted to give you a certain taste of some of the terminologies which are being used by to regulate the circadian clock. So now, so as we know that most of the organism, most means majority of the organism on this earth, their physiology as well as the behavior is nearly regulated by the sleep-wake light dark cycle of the earth, which is close to 24 hour, not exactly 24, close to 24. Somewhere it is 24.5, somewhere it is 23, 23.4 or 7. So it is close to 24 hour. So many of these daily rhythm they persist with an intrinsic close period. So apart from the outer 24-hour light dark cycle, many of the daily rhythms in our body as well as outside, they have the close periodicity of 24-hour, even when we place the animal under constant condition. What does it mean? Like suppose we are regulated or synchronized our activity to the outer light dark cycle. So when it is night, generally we come to our house and goes for sleep, but during the daytime we starts working, we starts doing our daily activity. That means our activity is being regulated by the outside external cue or stimulus, that is the 24-hour light dark cycle. But when we place the organism under constant condition for several days, even though we are being separated from the outside world, outside LD cycle, even then also our body clock starts synchronizing or starts moving and synchronizing the other physiological and biological processes in our body, which is also close to 24 hours. So as I showed you in the previous slide, that the circadian clock, it is mostly resetted or affected by several environmental cues such as light, food, drug, sound, etc. But light is considered to be one of the most potent or important Zeitgeber. Zeitgeber is a term, it's a German term which is used for stimulus or a cue. So why circadian rhythm is important? Because circadian rhythm, it's a 24-hour phenomena. So it regulates our internal physiological, biological and metabolical as well as the behavioral processes to the external 24-hour light dark cycle. So that's why in our tradition also, like, it has been said that you get up early in the morning with sun rising and then sleep in the night proper time, so that your rhythmicity will be maintained. You will you will perform or you will do your work with maximum efficiency. So we are going to see all these things in my uh, research. So so if this circadian rhythm get disrupted, so what happens? So when the circadian rhythm gets disrupted, as I told you, like melatonin and other physiological functions, they all get disturbed. So once we start means becoming awake, doing work at odd times of the day or night, then all our body uh, physiological, metabolic and neurological functions get disturbed, and that leads to sleep disorders, hypertension, diabetes, cancer, and in present topic I'm going to discuss about the cognitive deficit. So cognitive deficit is a state of mind during which we our senses are working properly, we are able to judge things properly and do things properly. So when the circadian rhythm disruption occurs, there is cognitive deficits. So it has been regarded by International Agency for Research on Cancer that shift work or jet lag may cause circadian disruption, and it acts as a carcinogenic agent to human being. So recently we found in some European countries that the nurses who were working late in the night, they were diagnosed with breast cancer because there was disruption of their circadian rhythm in the body. So in my present research, I'm going to use one of the Ayurvedic drug known as the Bacopa moneri or Brahmi, and it has been extensively used for treatment of anxiety, improving the learning and memory for several centuries in our country. So, and the pharmacological properties of Bacopa moneri is mainly because of the presence of one's key important ingredients called the saponins, and that is a bacosides. So in bacosides also there are several bacosides, and so I have taken one specific bacoside for my research, and it has been shown that the bacosides have neuropharmacological role in treating the disorders like insomnia, depression, psychosis, epilepsy, and it has been extensively used to cure these disorders. However, till date, even though Bacopa moneri or Brahmi has been extensively used in our society, but its role as a chronobiotic molecule. What does chronobiotic means? Substance or a drug which can regulate the circadian clock has not been studied. So I have studied the effect of Bacopa moneri or Brahmi or bracoside as a chronobiotic molecule to treat cognitive deficit in mammals. So the objective was to induce circadian disruption by creating an environment or simulating chronic jet lag shift work condition. And how we do that? either by advancing the LD schedule, light dark schedule. Suppose the light every day the light is on at 6 a.m. in the morning and off at 18 hour, means in the 6 in the evening. Now what I will do to simulate the jet lag shift over condition, I will advance the lighting schedule. Instead of getting on at 6 in the morning, I will put it on around I will advance 8 hours, so it will be on around 10 in the night, 10 hour in the night, means 10 p.m. in the night. That means you have advanced the circadian clock or advanced the stimulus. So the circadian clock will try to match with the external light dark cycle. So it will take some time. So this is the way I have simulated the chronic jet lag shift work condition in my experiment by advancing the LD schedule by 8 hours for 3 consecutive cycles. So, and then I wanted to see whether the Bacopa moneri extract or the bacoside is having any effect, or it is having any phase resetting effect on the circadian clock or not. So for that, what I did, we took adult male mice of AKR strain, nearly 25 to 30 gram, and then these mice were maintained into 12-hour light and 12-hour dark cycle under a constant environmental regime. Constant environmental regime is very important because in the previous slide I showed that it is the temperature also which can reset the suprachiasmatic nuclei. So that's why in circadian biology we used to perform our experiment under constant laboratory condition to negate the effect of external environment on the output of our results. So what I have done, so we maintain the mice under 12-hour light and 12-hour dark, and we have provided food ad libitum to the mice. Then we randomly divided the mice into 5 groups, and each group of mice, in each group there were 5 mice. First mice group was known as the non-treated control. We didn't give anything to them. Then the second group is Beckel treated, in which we had only 20% gum acacia and 2% twin 20, and 500 microliter per kg body weight was injected. In the third group, 40 milligram per kg body weight of bacoside was injected. In the fourth group, 80 milligram per kg body weight, and in the fifth group it is the 80 milligram per kg body weight. And this treatment continued for throughout the experiment, even after in group 3 and 4 we stopped the treatment after 28 days, but in the fifth group we continued the experiment till our experiment got over. So we wanted to see whether which group is having the maximum or optimal effect in regulating the circadian clock. So generally what we did, we gave the bacopaside through oral gavage needle, and this was the setup we used for recording their locomotor activity rhythm in mice. So you could see this is a wheel, and this wheel is connected to a sensor. When the wheel rotates, there is a sensor which senses the movement of the mice. So whenever the mice runs, it starts giving its activity in the form of actograms. So the dark band you could see is the activity of the mice during the night period, because mice are actually mammals. Are two types of mammals are there, nocturnal and diurnal. Another is also there, crepuscular, which, so what does nocturnal means? Those animals which are active during the night time, and the diurnal means those animals which are active during the daytime. So we human, we are active during the daytime, so we are diurnal animal. But like the cockroaches, the mice, rats, they are mostly active during the night time, so that's why they are called nocturnal. So when you see their activity, they will be maximally active during the dark time. So you could see the light and dark band. This dark band denotes the activity, the light bands denote the rest. And So each row represent 1 day, and so we used to record the activity for several days and months. So what we did, after acclimating the mice into 12:12 hour light dark cycle, we transferred the mice into constant condition. Why in constant condition? Because we wanted to uh, uh, see that the external environment does not affect the output of the means the biological clock. So that's why we place the mice under the constant condition with 12-hour light and 12-hour dark, and the mice were given continuous access to the running wheel so that they can perform their activity whenever they want. That is, voluntary locomotor activity can be recorded. So after the baseline recording of the mice for 10 to 12 days under 12-hour light and 12-hour dark, so the light was on at 6 a.m. in the morning and off at 18 hour in the evening. So after the stable 12:12 hour light dark cycle, the mice were subjected to chronic jet lag shift work condition. I'll show you in the next slide. So during the jet lag shift work condition, what happens? As I told you, we used to shorten the light schedule, or we used to advance the light schedule by 8 hours. So when initially the light was on at the 6 in the morning, now it is going to get on 10 in the night. That means we have advanced the lighting schedule. So the animal will be confused because they were expecting the light to be on 6 in the morning, but it got on quite early. So then they will start feeling that now it's time to go to sleep, because once the light gets on, being nocturnal, they stop moving. But it will take some days for them. Such kind of we experience such kind of events when we move from India to USA. It will take our body to get synchronized to the USA condition within 3 to 4 days. Someone takes 8 to 9 days. So that time taken by the body to resynchronize or entrain to the new light dark cycle is known as entrainment or re-entrainment. And the time between the previous and the new cycle is known as the transient cycle. So what we did, we used to advance the LD cycle after every 3 days. So there were three consecutive cycles. So after third consecutive cycle, we moved back again to our original cycle. So you could see in this diagram, this is the simulation of jet lag shift work condition. So this white bar shows the light time, that is when the light is on 6 in the morning, and 6 in the evening the light gets off, and at the same time this is the dark condition. So you see the animals get active during the night condition because they are nocturnal. So for keeping the animals 10 days under 12-hour light and 12-hour dark cycle, their endogenous clock, their inside clock, the biological clock get synchronized to the 12-hour light dark cycle, and we will see such kind of activity rhythm. But when we advance the LD cycle by 8 hours, the mice are confused and they will take some time to reset to the next 8 hour, means new lighting schedule. So this time is known as the time taken by the animal to resynchronize to the new LD cycle is known as the transient cycle. That means during this transient cycle, all our physiological function or physiological, biochemical and behavioral processes are disturbed. And after some days, then in some animal, they will re-entrain much faster. They will take 3 to 4 days. Some will take many more days. So then the animal will be synchronized to the new LD cycle, and we call it as re-entrainment. So the word entrainment means synchronized, synchronize to the environment. Re-entrainment means to resynchronize to the new environment which we have changed. So this was the protocol I use. So I'm just talking about some of the techniques which I use to monitor the cognitive deficit in mice. So first was the locomotor activity by simulating jet lag shift work, as I discussed here. Then to find out whether the animals were experiencing cognitive deficit or not. So what we did, we uh, to study cognitive deficit, there are several parameters, behavioral parameters which we use. So one was the Barnes maze, which I use for my experiment to study the cognitive deficit in learning and memory during the simulated jet lag or shift work condition. And I studied both the short-term memory and the long-term memory. So how the jet lag causes the cognitive deficit and whether it affects our short-term memory and long-term memory. So what we did, first we adapted the animals, and then what we did, we allowed the animals, we trained the animals. So the animals will were trained for 4 trial per day, and every trial there was 60 minutes of gap during the all the 4 days. Then we recorded the memory on the final day, that is the probe day, or the that is known as the probe trial day. And during that day we record the reference memory, that is the short-term memory and the long-term memory. So for recording the short-term memory on the 5th day, we place some cue or inside. So first of all, I will show you, this is the Barnes maze. This is a table with different types, different holes, and in one hole we have placed the cue, that is either food. So what we do, we place the animal here in the center, and then the animal will start searching for the cue. So if it is mentally sound and if it has learned, then it will reach the target much faster. But if it shows the cognitive deficit, it will take long time to search, or it will do a lot of error by moving to other location rather than their main target hole. So that we use to record in our camera, and then we use to count the time taken by the animal to reach its target hole. So what we did on the 5th day, the target hole was, we closed the target hole because we have trained them, and so we have trained them to find the target. So on the 5th day we closed the target hole, and the probe trial was done to determine if the animals remember its target goal or not, or where it is located. So it is a special we wanted to see whether its special memory is working properly or not. Then the number of pokes or the error in each hole and the time taken to reach each hole are measured. Then what we do, after the 5th day, we try to measure the long-term memory, and that we measure on the 12th day without training in between, like from 5th day to 12th day. So 3 type of categories, searching categories the my uses. First one is the direct, that is it uses the special cues, its surrounding, to move to search that target hole, or it will go to its adjacent hole, but it will go in this quadrant rather than moving if it is what do you call normal. But when it is not normal, then what we will, it will try to try to find the normal hole, but it will move to different holes in both clockwise and anti-clockwise direction. That means it will take a lot of time. So during the spatial or that direct searching strategy, it will move directly to the target hole. During the mixed strategy, what it will do, it will cross the center here, and it will unable to recognize or organize how it should move. In the serial strategy, what it used to visit the target hole preceded by the first to adjacent. That means this is the target hole. First it will move here or here, and then it comes to know, no, my main target is here. So it will take some time. So we used to measure that time, how much time this animal is taking. So another test we used was the noble object recognition test. During this test, what we used to do, if the normal animal or the mouse or the rodent, they have the ability to explore the noble object. So whenever you place a new thing in the arena or the place, the mouse has ability to explore that. So but when they are cognitive deficit, or when they are not in the normal frame of mind, then they lack the motivation to explore those things. So we have used this test after simulating this circadian disruption by simulating jet lag shift work condition, to find whether their memory is, or hippocampus is functioning properly or not, so that they are doing their work properly or not. That means we wanted to check the episodic memory. So for that we use the noble object recognition test. So first what we did, we placed, as you see, a rectangle boxes. So first we removed everything, nothing was there. So first we habituated the animal by placing the animal inside this rectangular boxes, and so that they will start exploring that arena or the area. After that, what we did, we again placed the mice in the boxes, but we placed some new objects in those area, and we kept it for 5 minutes. So once they are trained inside the box, what we did, we removed those object. I mean, those from those two objects, we placed one object which was similar in the last training, and then another new object was placed. Initially, you see here the two cylindrical uh, objects are there, but in the what you call familiarization, we used to place one cylindrical and another is the cubical form. So what the mice will do, it will go to the cylinder, but since it has already known that, it will try to move immediately to the cubicle, because it's a noble object, sorry. So the mice has ability to explore, so it will spend a lot of time in exploring the cubicle object. So this, we want to see how much time it is taking to explore the noble object. So what we used to do, we used to keep the animal for 3 minutes inside each what you call the box, and we used to see through our video recordings that how many times the mice is using its paws, its nose, its mouth to explore the object. So this we known as the exploratory behavior. So the probe test or the test phase, in test phase, what we used to calculate, we used to calculate the discrimination index. So what is discrimination index? So it is basically whether the mice is able to discriminate between the noble object or the familiar object. Because as I told you that in noble object, the mice has the ability to explore the new object, but when it is not active, when its mind is not functioning properly, it will be confused, and instead of exploring the new object, either it won't go anywhere, it will be sit at the side of the wall, or it won't have motivation to explore. So this discrimination index is calculated as the time spent with the noble object minus time spent with the familiar object, multiplied by 100 upon total time spent for both noble object as well as the familiar object. So if the value of the discrimination index is positive, that means the mice is performing properly and it is having the preference for the noble object. Whereas when the discrimination index is like 0, that means it is confused, it is unable to decide, or it is unable to take decision. That means it has cognitive deficit. And the negative discrimination index indicates that it has preference for the familiar object, which is not the property of the rodents, because if the rodents are normal, they will explore the new objects. So, hello. Are you able to hear?
Yes, sir.
Okay. So now I'm coming to the my results. So these are all these chart. What you see, it is an locomotor activity chart of the mice, the true locomotor activity chart, and the top you could see the black and white solid bars. So black and solid bars, the black bar represent the dark period for the animals, and the white bar represents the light period. So this is the double plotted actogram. The activity is 2 times plotted. So why it is 2 times plotted? Because we wanted to see the clear cut picture. So that's why we used to plot the activity twice. So initially what we did, initially the animal, the light was on at 6 in the morning and off at 18 in the evening. So just when the light goes off, the animal starts becoming active. So we, as I described, that we kept the animal for 10 to 12 days under the LD cycle, light dark cycle. So their activity was synchronized to the light dark cycle. Synchronized means entrained. So you could see the clear cut pattern of rest and activity here. So once they were entrained, or their activity was stabilized to the external light dark cycle, we shifted the light dark cycle by advancing it to 8 hours. So instead the light is on means off around 6 in the evening, it and on 6 in the morning it becomes on. You see here it becomes on around what you call much more earlier. That means close to 10 in the night. That means now the animal start thinking that now the light has off, so let's start moving. So the mouse will start moving during the irregular time. So their activity is not settled. So what we did, we we repeated this cycle for at least 3 times, 8 hour, 8 hour, and 8 hour. We kept the animal for 8 hour advance for three days, and then again we advanced their LD cycle for 8 hour for 3 days, and again advance. So it on the after the third cycle it came to the normal LD cycle. So you could see on the top normal LD cycle and at the bottom normal LD cycle. So use so what we recorded through the locomotor activity, we could see the whether the animal in the endogenous clock period is 24 hour or not. So this was the, you could see here that it is showing the periodicity of the animal is 24 hour. But during the jet lag condition, the periodicity or the endogenous period is less than 24 hour and it is 21.8. It is very deleterious to the animal, or even to us also. It should be close to 24 hour, but this is 21.8. And after we left the animal to the again normal condition, it resynchronized to 24 hour, but it took lot of time to resynchronize to the normal LD cycle. So we performed the experiment with many animals, and you could see the pattern of this is the chart for first animal, second, third, fourth. I want to show you because you could see that they are active during the night time and taking rest during the daytime. But after the advancement of the LD cycle, their activity gets disturbed, and it takes lot of time to resynchronize to the new LD cycle. And this is highly deleterious. During this time, the mice are confused, and they are unable to perform their task and activity properly. So this is the endogenous period. Endogenous means within the period of the our clock, our brain clock. So during the pre-jet lag condition, that is before advancing of the lighting schedule, as you saw here, this is the pre-jet lag condition before shifting of the light dark cycle, and this is during jet lag condition. The endogenous period has highly changed, and this is the post jet lag condition, that is after it was resynchronized to the previous or original cycle, it regains its 24-hour periodicity. So you see during the pre-jet lag cycle for the 6 animals, their endogenous period was 24 hour. That means their clock is functioning according to the LD cycle. But as we disturb the circadian clock or by simulating jet lag shift work, what happens? You see the their endogenous period is highly disturbed. It is less than 24 hour, 21.9 and 22.8, so on and so forth. You could see that means the period of the circadian clock is highly deviated or disturbed during the shift work condition. Also, you could see the activity profile. So the activity profile, from here you could see no activity or very feeble activity. That means the animal were during the lighting condition, they were taking rest. But as you see around 18 in the evening, they all become active. This bar shows the activity. You see the peaks, this is the activity. That means the animal are active during the night time. But during the jet lag condition, you see during whole day they are disturbed. They don't know whether to be active at what time. So this will lead to lot of disturbances. And in nature, suppose if they are living in the jungle, they can be easily predated by their prey, so they can easily be killed. So that's why normal functioning of the circadian clock is very essential, very important, not only for the animals but even for the human beings. So some of the parameters which I studied, the circadian parameters was number of transient. As you remember, I showed you in my initial slides that the transient is the time taken by the circadian clock to resynchronize to the new shifted LD cycle. So the control animal which was not given any drug, it took nearly 16 days to resynchronize to the new LD cycle. Or so the number of transient were more than 15 days in control animal, means untreated control. In vehicle also it was very high, but in what you call, you see the group 3, that is the 40 milligram per kg body weight of the Bacopulmonary, 80 milligram per kg and 80 milligram per kg continuous, the number of transient is statistically reduced. And another parameter which we study was re-entrainment, means how much time it takes to resynchronize to the new LD cycle. You can see that both the onset of activity and the offset of activity. So onset of activity starts when the light gets off, and the offset means the activity stops when the light is on. So you see that the both onset and offset of activity takes longer time in untreated animal as well as the vehicle treated animal, but it is statistically reduced. The number of days is statistically reduced in what you call the animal with which are treated with the 40 milligram per kg body weight of bacoside, 80 milligram as well as the 80 milligram continuous treatment. But in the graph you could see that 40 milligram compared to 80 is quite better. So another the factor which we studied about the alpha means the activity period. So you see that the activity period during the jet lag condition, pre-jet lag condition was close to 11 hours or 12 hours. But during the jet lag condition, it drastically reduced or statistically reduced to less than 10 hours. And in the post jet lag condition also it is reduced. Why? Because actually in post jet lag condition, though the activity has synchronized, but the duration, the time of the activity is still reduced. That means the body clock is still not rebounds to the initial condition. You could see here the period. I have shown that the period of the endogenous clock during the pre-jet lag condition is close to 24 hours. During the jet lag condition, it is less than 24. It will be around 22, 21. And during the post-jet lag condition, it again bounces back to 24 hours. So the bounce phase result I wanted to show here. So this Latency we have measured it in seconds. So latency means the time taken to reach its target hole in the pre-jet lag condition was 2 and below that. Means in some animals it was much below as we trained the animal. So you see here, like day one, on the first day of training, it took more time to search its hole. But as we train the animal, the time starts decreasing, and on the fifth day, on the trial day, it decreased further to less than 1.5, and on the 12th day it was close to 1.5 and below that in the pre-jet lag condition for all the control as well as the treated animal. So you see here, during the jet lag condition, the Bacopa moneri treated mice were their state of mind were much better compared to the vehicle and the untreated control. They took less time to reach their target hole. That means the Bacopa moneri has helped them to remember or their keep their cognitive ability intact. They were not disturbed, or though they were disturbed, but they were not as much disturbed as you could see in the jet lag animals. So the time taken by the jet lag animals were comparatively or significantly low compared to the vehicle and the untreated control for both the short-term memory as well as the long-term memory. In the noble object recognition task, we found that the exploratory time for the control means for control and the vehicle treated animal, they explored less. That means they were not interested. That means their mind or their brain, they showed cognitive deficit because of the jet lag condition, or and then the vehicle treated or what do you call, the drug treated animal, they explored more. That means the Bacopa moneri has helped to improve the cognitive deficit which they experienced during the jet lag condition compared to the control and the vehicle animal. So from this, what we get, we get the idea that the bacoside or the Bacopa moneri extract has the phase resetting property. Phase resetting property means that it has ability to change the phase of the disrupted clock. So this property can be used to treat various circadian disorders which occurs during the shift work or jet lag condition, which we experience during the our transmeridian flight when we move from one country to the another country or faraway country like the US, Japan, and or when we do work during the different hour of the light dark cycle, or the person who are working in the hospital or in the industries or in the army. So this bacoside can be used as chronobiotic molecules. Why? Because we saw that it has significantly reduced the number of transient. It also reduced the what you call the percentage of uh, uh, the animals performing better during the jet lag condition. And also we found that compared to the onset of activity, offset of activity to resynchronize took longer time. So these are all suggest that even though the animals show normal activity, still their inner clock is not stable. It will take much more time to stabilize. So our behavioral tests also confirm that during the chronic shift work jet lag condition, there is loss of memory or the cognitive deficit occurs. But when we treat the animals with the bacoside or Bacopa moneri, it boosts the memory, learning and memory, and preserve the cognitive deficit. So we want to, as I described before, that it has the phase resetting property. So how we could say that it has phase resetting property, as we saw in the jet lag shift work experiment. But to prove that it has a phase resetting property, one key or important features or the experiment we do in circadian biology is to place the animal under constant lighting condition or constant dark condition. So since mice is a nocturnal animal, we used to place them under constant darkness, because for them dark is the normal cycle for them. So under dark, they can easily move. But lighting condition is aversive behavior. They used to hide from light. So that's why we place the animal during the constant condition. And you could see on the top, I have placed the black bar, a continuous black bar. There is no white and dark. So continuous black bar shows that the animal is placed under constant dark condition. So when we place the animal under constant dark condition, instead of straight line, their activity starts slanting or advancing each and every day. So this phenomena is known as the free running condition. So when we place the animal under free running condition, they start advancing each day, and it is known as the free running condition. So after 10 to 15 days of free running condition, what we used to do on the 10th or 11th day of the stable free running condition, you could see I have drawn the red line, means they are in a same inclined plane. So on the 10th day, the arrow denotes the day on which we used to give the dose of the Bacopa moneri, and this is the different time. So what we did, we gave animal bacocide at different time of the day, and we want to see whether the bacocide is having any time dependent effect or not. So initially we gave the animal the bacocide at this time point. It is called CT0. Then at this time point it is known as CT3. This is at CT6, and you could see the red and the blue line. There is very less difference between the red and the blue line. They are nearly parallel or touching each other. But as we start giving bacocide at different time point of the day, there is difference in the orientation of the red and the blue line. So after giving the doses for the animal on the 10th day, we used to leave the animal for another 10 to 12 days to see whether it has bacocide has changed its phase or not. So you after the 10th or 12th day, what we did, we got this activity chart of this animal, and we found that their activity has shifted little backward. That is, it has caused the phase shift in the backward direction. This is known as the phase delay. But at the CT6, CT means circadian time. Circadian means when the animals are placed under constant condition. So when we have kept the animals under constant condition at CT6, at CT6 time point, their clock is highly advanced. You could see CT6 is very big advance, and this advance is close to more than 2 hours, which is a huge. And at CT9 again you could see the advance. So at CT12 you could see the phase delay, CT15 huge phase delay, and then again less phase delay at CT18 and CT21. So what we did, we plotted the value of all these phase shift at different time points, and we draw a phase response curve for the bacocide, and we found that these phase shifts were plotted along the x and y axis at different time point. So this is the CT0, CT3, 6, 3 hour apart for complete 24 hour for all the animals, and this is known as the circadian time. And this is the phase shift, that is the difference between the initial phase and the after giving the bacocide, how much it shifted the clock. So when it was plotted, we saw that when we give the bacocide in early, means the late night, the clock is showing the maximum phase resetting effect. That is, the bacocide is shifting the clock when it is taken during the late night. So that means taking the drug or the bacocide shows time dependent effect. So if we take the bacocide at any time, it won't show its effect. So if you take the bacoside around 9 o'clock or 9 circadian time or the 12 circadian time, the effect is not too much. But when you take at the late subjective night, that is around 1 o'clock in the night, or early subjective night like 10 or 12 in the night, it shows the maximum effect. So to further substantiate whether it is, whether it is really having that effect or not, we perform the pharmacokinetic study of the bacoside. And here we took, instead of mice, we took rats. Why rats? Because we have to draw bloods at different time points from the same rats. So we wanted to see how the drug has metabolized. So one branch of biology which study the effect of time on the dynamics of the drug is known as the chronopharmacokinetics. So here we study the chronopharmacokinetics of the bacoside. And so what we did, we took the animal, that is the rat, and gave them what you call the oral and the intravenous injection of bacoside at 2 time points, that is the ZT3, Zeitzeberd time 3, and ZT19. So here keep in your mind that ZT3 is, I told you initially that we used to put the light on around 6 in the morning. So 6 in the morning is known as ZT0. So ZT3 means 9 in the morning. So we performed the one experiment at ZT3, that is 9 o'clock in the morning, and another experiment at ZT19. So ZT12 is known as the time at which the light is off, that is 6 in the evening time. But here we are performing the experiment at ZT19. That means after 12 we take 7 hours more, that it will be close to 1 or 2 o'clock in the night. And we wanted to see whether to see the effect of bacoside on the 2 time point. That means that at which time points, if we take the bacoside or Bacopa moneri, it will show its maximum effect. So and also we wanted to see the routes of ingestion, whether taking orally through mouth is beneficial or giving through the blood it is beneficial. So we took the rats, divided into 2 groups. One is ZT3 groups and another is ZT19 group, and we gave the oral doses of bacoside 100 milligram per kg body weight, 5 mice each, and intravenous we injected the bacoside in there through the tail vein, and then we sampled their blood. We took 250 microliter of blood at 0.25 hours after giving the dose. The moment we give the dose, after 2.5 hour, we start taking the blood at 2.5 hour, 0.25 hour, then 0.5 hour, then 1 hour, after 1 hour, then after 2 hours, 4 hours, 6 hours, 8 hours, and 24 hours from the same animal. So we used to do the blood sampling from the same animal for complete 24 hours. We want to see the effect of how the drug is showing its pharmacological effect. And then this blood was processed and HPLC was performed to see the the expression pattern of the bacoside. So after that, we just a minute. So then what we did, we, after doing the HPLC, we analyzed the data and the various pharmacokinetic parameters we saw or we measured. that was the dose, the maximum concentration of bacoside in the blood, then the time at which it shows its maximum effect, then the area under curve, then the T half at the time at which it was half time at which it shows its maximum effect, clearance time, the bioavailability, how much it was available, and relative oral bioavailability. So we compare here the ZT3 and ZT19, and we found that compared to the oral and intravenous, it is the oral which is the best or effective way of taking the bacoside. And you see that the maximum concentration of oral was 380.65 nanogram per ml, but at ZT3, but at ZT19 it was 3.5 times or close to four times more uh, uh, bacoside was available. That means if we take the bacoside late in the night, it will show maximum effect, or it has maximum availability, and when it is maximum available, it will show its maximum effect, as we saw in the phase response curve. So in the phase response curve, you saw that the maximum effect or phase shift occurred during the late subjective night around CT3. Also the area under also the T max hour in at ZT3, it takes 8 hours after a dose to show its maximum effect, but when the drug was given at ZT19, it shows its effect within 6 hours. That means it takes 2 hours less time to show its effect. So the area under curve for ZT3, you see it takes 638.35. Here it is nearly double. So it shows maximum effect, or its availability is maximum. Availability of bacoside in the body is maximum, or 2 times more at when we take the bacoside at ZT19. Also the bioavailability, that is the available bacoside in our body, when we take at ZT3 it is 41%, but or 41 time percent, but here at ZT19 it is 94% available. And the relative oral bioavailability, when we compare with ZT3 to ZT19, it is more than 200% at ZT19. So this shows that bacoside, if we take at a specific time point, it shows its maximum effect. So this is known as the chronopharmacology or chronopharmacokinetics of the bacoside. So in this pictorial diagram, I have shown you my graph that the plasma concentration of bacoside availability in nanogram per ml at ZT3 and ZT3 intravenous and oral. You see that the oral, the bioavailability is more, but relative compared to 3 and 19, the relative or bioavailability is much more higher, and it occurs much earlier. Like it takes 6 hours, here it takes 8 hours. So the time of taking the drug is very important. This 9 shows ZT3. You see the red arrow, it is ZT3, where we give the bacoside and we start sacrificing the animal in ZT3 group. And this shows the ZT19 animal, that is close to 1 o'clock in the morning or 1 o'clock in the late night. It when we take bacoside at this time point, its efficacy is much more. So if we give bacoside to the cognitive deficit animal at late in the night, it will show its maximum efficacy. It will enhance the learning and memory much more in a better way compared to the animal which are given the bacoside or the bacapamenary early in the morning. So we found the expression of initially I showed you the period and the clock genes, and we found that the expression of the clock genes at ZT19 late in the night were comparatively much more expressed. That the protein were much or highly expressed in these what you call, this is the suprachiasmatic nuclear region, and this is the third ventricle. You see the stained area. So you see here that the expression pattern of different clock genes, that is the BMAL, the PER1 and the PER2 and the clock, is much more compared to that in the controlled animals. So my result, I'm summarizing my result that the circadian disruption as a result of cognitive deficit or jet lag shift as a result of jet lag shift work causes cognitive deficit in mice, and bacoside can be used as a chronobiotic molecule to ameliorate the both short-term and long-term memory deficit as a result of desynchronized or destabilized or disrupted circadian clock. Hence, it can be used as a chronobiotic molecule similar to the available chronobiotic molecules in the market, which are melatonin, serotonin, 5-HTP. So, and since this is a natural bacoside or Brahmi, which is used from million and million of year or ages in our society, in Indian society, it is much more eco-friendly or beneficial to use Brahmi as a chronobiotic molecule to treat the cognitive deficit or deficit in learning and memory as a result of desynchronized circadian clock. And it can be used as a, this shows, my results clearly shows that the bacoside has a therapeutic potential in resetting the circadian clock by re-accelerating or resynchronizing the disturbed rhythm to the original LD cycle. So it may help in ameliorating the cognitive deficit and can be used for clinical management or clinically managing the sleep-wake disorders by and it may help in increasing the efficiency of the people who are working late in the night in the industries, in the hospitals, in the army. Because of that, their clock get desynchronized. So taking bacoside at right time in proper doses will definitely have the therapeutic potential in ameliorating the cognitive deficit in not only in animals, but it has been shown that it is also efficient in humans. But the chronobiotic effect no one has shown, and first time we have shown in our lab. I have done several gene work also, but as you saw, it took a lot of time, so I didn't present my result. So I would like to thank the project was funded by the SERB New Delhi and Department of Science and Technology to me through two true projects. And I would like to thank Department of Geology, Banaras Hindu University, and my postdoctoral department where I did my work is Department of Pharmaceutical Engineering and Technology, IIT BHU Varanasi, for providing me the lab facility where I carried out this work. And I would like to thank my mentor, my guru, Professor M. Singh, my PhD supervisor, who I'm having deepest regard, and still he is my both professional and mentor today also, Professor Sai Ram Krishnamurthy, sir. I'm really thankful to him for giving me the opportunity to work in his lab. And thank you to all my colleagues and members who have patiently listened to my talk. Thank you very much.
Thank you so much, sir, for enlightening the students about your neuroscience. And now I would like to call Ranjana ma'am to present over the memento of honor to sir.
Dr. Kumar, very nicely and enlightening us with your talk. Right.
Thank you, ma'am. Thank you very much.
Thank you so much, sir, for attending our event. As earlier in the session, I told about what IIC Galgotias University is and what we here provide to students. Here we promote IIC Galgotias University promotes research and innovation. Here we provide platforms to startup and provide them with all kinds of supports to make the startup be in the ideation stage or is in the revenue generation stage or in any stage in traction stage. So here I would like to tell you about one of our startup, Udyami. It is
Okay.
a semi audible.
Yes, yes.
Okay, sir. So we have a startup named Udyami where we have created they have created an ecosystem of entrepreneurs, mentors, investors, incubators, and support providers like bank, fund, vice chancellors also, and venture capitalists too. And your knowledge perspective and understanding of your field will help the entrepreneurs related to your domain, neuroscience, to excel in their field. And I would like to request you to kindly accept our invitation to provide your mentorship and be the part of Udyami as a mentor, as a guide to all the students, all the startups that are incubated and want to do something in the field of neuroscience.
Oh,
We would be
definitely.
delighted to have you amongst us.
Yeah, definitely. I'm thankful to your institute, and also I would like to tell all of you that here in Israel, like if anyone wants to do some research or do their PhD or anything, um, talk to Gaurav sir. I'm always there to help you all
Okay, sir.
in
So,
all the
if
aspects.
possible, if you can drop down your email ID in the chat box so that we can connect with you.
Yes, definitely.
Thank you so much, sir. We'll connect you with you regarding Udyami, and it will be our honor that you will be the part of Udyami.
Yeah, yeah. Thank you. Thank you very much.
Thank you so much. Thank you so much, everyone, every faculty members, our speaker, and our dear students for attending the event. I will be sharing the feedback form in the chat itself. Kindly fill it.
So, can I leave now or I still have to wait?
No, sir, you can leave.
Uh, is there anything else? Gaurav ji is there? Dr. Gaurav?
Actually, sir, due to some network issues, he's not able to join now.
Okay, okay, okay.
Yes, sir.
So,
Thank you
so
so
I'm,
much, sir.
yeah, yeah. Okay. Thank you very much. Bye bye. And best wishes to all of you.
Thank you, sir. I will be sharing the feedback form in the chat itself. Kindly fill it. The feedback form will be open till 3 p.m. So, if possible, everyone please fill the form before that. Over to you, ma'am. Actually, your voice is not audible. It's breaking. ma'am. If you could please rejoin. Still, we are not able to hear you, ma'am.
Some network problem.
Yes, ma'am.
Hello. Ma'am, what is fill-in specialization?
Specialization, if you are doing any specialization in the domain, right? Like in BTech, we have in computer science, we have cybersecurity, AI/ML, like in that.
No. I am in first semester BSc MLT.
So, write there NA or none.
Okay. Okay. Thank you.
Okay. Hello, Ranjana ma'am. Sorry, ma'am, we are not able to hear you now also. There must be some network issues.
So if you